Oxygen once nearly wiped out all life on Earth. By the end of this video, you will understand how the very substance that sustains us first acted as a planetary poison, and why that catastrophe unlocked complex life. How did a waste product from primitive bacteria trigger a global extinction? And why did this same toxic gas later become the breath of animals? What would our world look like if oxygen never became our ally? Oxygen was not always a friend to life. In fact, it began as a poison, a waste product that nearly wiped out the entire living world. This is the central paradox of our planet's history. The same element that now fills your lungs with every breath once suffocated and destroyed Earth's earliest inhabitants. To understand how that happened, we have to go back more than 2 billion years to a time when the atmosphere contained almost no oxygen at all. The first life forms were simple, single-celled microbes that thrived in an oxygen-free world. They fed on chemicals from the oceans and volcanic vents, and they released a tiny byproduct into the water. That byproduct was oxygen, and at first it was harmless. But as these microbes multiplied across the ancient seas, their waste began to build up, and that buildup triggered the greatest crisis life had ever faced. The arrival of oxygen was slow but relentless. Over hundreds of millions of years, the gas accumulated in the oceans and then started leaking into the air. For the dominant organisms of that era, oxygen was a corrosive poison. It reacted with their cellular machinery, tearing apart essential molecules and causing widespread damage. Entire ecosystems collapsed. Species that had ruled the planet for eons were pushed to the edge of extinction. This event, known to scientists as the Great Oxidation Event, was a chemical war on a planetary scale. It reshaped the atmosphere, transformed the oceans, and left only a handful of microbial survivors that could tolerate or even use oxygen. It was one of the most devastating mass extinctions in Earth's history, far worse than the one that would eventually eliminate the dinosaurs. But here is where the story takes a surprising turn. That same catastrophe that wiped out so many ancient life forms also cleared the stage for something entirely new. Oxygen, once a poison, became a powerful source of energy. The few microbes that learned to harness it gained a huge advantage. They could extract far more energy from their food than any oxygen-free organism could. That extra energy made it possible to build larger, more complex cells. It opened the door to multicellular life, to animals, to plants, and eventually to us. So the very crisis that nearly ended life on Earth ended up creating the conditions for complex beings to emerge. It is a war we have been living with ever since. And in the next part of this journey, we will see exactly how those first oxygen-breathing pioneers managed to survive and thrive in a world that had been poisoned by the very gas they now depended on. Before oxygen, Earth was a very different world. The sky was not blue. It hung in a hazy orange tint, thick with methane and ammonia and volcanic ash. There was no ozone layer to block ultraviolet radiation, so the surface was bathed in high-energy light that would be lethal to most modern organisms. The oceans were warm, rich in dissolved iron and sulfur, and they churned with chemical reactions instead of waves stirred by wind. This was a planet without free oxygen, a world that would feel alien and suffocating to us, yet it was perfectly suited for the first pioneer of life. Those pioneers were tiny, single-celled microbes known today as anaerobes. They did not breathe oxygen because there was none to breathe. Instead, they fed on the chemicals around them: hydrogen, carbon dioxide, sulfur compounds, and dissolved metals. They used these raw materials to build energy, a process called chemosynthesis. To us, that might sound slow and inefficient, but in that ancient environment, it was the only game in town. These microbes spread across the seafloor, forming thin mats that blanketed the shallows and the deep rocks. They were simple, but they were survivors. The atmosphere itself was a mix of gases that would be toxic to us: methane, ammonia, carbon dioxide, and nitrogen. Trace amounts of oxygen might have appeared briefly from sunlight splitting water molecules, but it was quickly consumed by reactions with iron and other elements. There was no reservoir of breathable air. The oceans were stained green and rust-colored from dissolved iron, and the shoreline was nothing but bare rock and sand. No plants, no soil, no green at all. The continents were barren, baked by radiation from a young sun that shone about 30% dimmer than today. For hundreds of millions of years, this quiet chemical world hummed along. The only sounds were the hiss of volcanic vents and the gentle lapping of anoxic tides. Life was small, simple, and stable, but that stability would not last. Inside those microbial mats, a tiny minority of bacteria had discovered a new trick. They could split water molecules using sunlight, a process that released a waste product they had no use for. That waste was oxygen, and at first it did not seem like a problem. The oceans and rocks absorbed it eagerly, reacting with iron to form rust and sinking to the seabed. The planet had a natural buffer, a way to neutralize this new toxin, but those microbes kept multiplying. They kept splitting water, and over millions of years, the buffer began to fill up. The iron turned to rust and settled, and there was less and less room for oxygen to be absorbed. Slowly, ever so slowly, the gas began to accumulate in the water, then in the air. What started as a harmless byproduct became a silent, creeping poison for the vast majority of life that had evolved without it. The original Earth, a world built around chemical energy and anaerobic survival, was about to face its greatest test. And the microbes that had thrived for so long had no idea what was coming. That ancient world was built from a very different mix of gases. The sky above the young Earth was not filled with the oxygen we breathe today. Instead, it was a thick chemical stew dominated by nitrogen, carbon dioxide, methane, and vast amounts of water vapor. There was virtually no free oxygen at all. If you could have stepped onto that shoreline, your lungs would have felt nothing but poison, and you would have collapsed within moments. But for the first anaerobic microbes, this atmosphere was a warm and welcoming home. The gases came from two main sources: volcanic outgassing and asteroid impacts. Deep beneath the crust, volcanoes belched massive clouds of carbon dioxide, sulfur dioxide, and hydrogen. These eruptions shaped the early air, adding layer after layer of reducing gases, meaning they could easily donate electrons and react with other molecules. Asteroids and comets also brought in organic compounds and water, smashing into the surface and releasing even more volatile chemicals. The result was what scientists call a reducing environment, the exact opposite of the oxidizing atmosphere we have now. In such a world, oxygen did not stand a chance. If any free oxygen did appear, perhaps from sunlight splitting water vapor way up in the upper atmosphere, it was quickly snatched up by eager chemical partners. Iron dissolved in the oceans would grab it and turn into rust. Free sulfur would grab it too. The planet was like a giant sponge, soaking up any stray oxygen before it could accumulate. This chemical hunger for oxygen is why Earth stayed oxygen-free for nearly 2 billion years. It is also why the early microbes, which lived by consuming these reducing gases, thrived. They had access to a steady buffet of hydrogen, methane, and sulfur compounds. They were perfectly adapted to a world where oxygen was an enemy rather than a friend. But that world was about to change slowly, invisibly, and irrevocably, and the very same chemistry that had protected them for so long would soon become their undoing. Deep in that ancient ocean, the first life did not eat or breathe the way we do. Instead, it fed on the raw chemistry of the planet itself. These microscopic organisms called anaerobes had no need for oxygen because they had never encountered it. Their entire existence revolved around a process known as chemosynthesis, a way of extracting energy directly from inorganic molecules. Think of it as a kind of chemical digestion that happens outside the body. They would latch onto molecules like hydrogen sulfide, a gas that smells like rotten eggs, and strip away electrons to build the energy they needed. It was slow, it was simple, but for them it was enough. These early life forms lived in sprawling mats on the seafloor, coating rocks and sediments like a faint, living film. They did not compete for sunlight because there was no sunlight to compete for. The oceans were dark and murky, and the surface was bathed in harsh radiation that would have destroyed any exposed organism. So they stayed below in the safe shadows, using the steady supply of chemicals that bubbled up from deep volcanic vents. Those vents were like natural energy stations, releasing hydrogen, methane, and sulfur compounds into the water. The anaerobes clustered around them, drawing sustenance from the planet itself. This lifestyle was incredibly inefficient by modern standards. A single microbe could produce only a tiny fraction of the energy that a later oxygen-breathing cell would make. But in that world, efficiency did not matter. There was no competition for these resources because nothing else could use them. The anaerobes had the entire chemical buffet to themselves. They multiplied slowly, generation after generation, filling the oceans with their tiny bodies. Over hundreds of millions of years, they changed the very chemistry of the seas, releasing waste products that would eventually become the planet's greatest challenge. But for now, they simply existed, and in existing, they quietly prepared the stage for a revolution. The energy they extracted from hydrogen and sulfur was meager but reliable. These first single-celled organisms, known to science as anaerobes, had perfected a way of life that did not require oxygen because oxygen did not exist. They fed on the chemical bonds of inorganic molecules, pulling electrons from hydrogen sulfide, dissolved iron, and methane that bubbled up from deep-sea vents and volcanic rifts. This process, chemosynthesis, was their only means of survival, and it worked beautifully in a world where oxygen was entirely absent. They also relied on fermentation, breaking down organic compounds without the help of oxygen, to generate the tiny amounts of energy needed to grow and divide. Their entire biology was built around an oxygen-free environment. Every enzyme, every membrane, every metabolic pathway had evolved to function in its absence. So when oxygen finally began to appear as a waste product from a new kind of microbe, it was not simply a foreign substance. For the anaerobes, oxygen was a direct and immediate threat. It reacted with the very molecules they depended on. It ripped apart their cellular machinery, destroying their ability to harvest energy. It turned their own internal chemistry against them. In other words, oxygen was a poison, a silent, invisible killer that attacked from the inside out. This might sound like a far-off problem, but it was about to become the defining crisis of the early Earth. The anaerobes had ruled the oceans for hundreds of millions of years, but their reign was about to end. The revolution had begun, and it would leave no cell untouched. But one group of microbes had discovered a revolutionary new power. They learned to harness sunlight directly, using its energy to break apart water molecules and grab electrons. This was a game changer. Instead of scrounging for hydrogen sulfide or dissolved iron, they could split the most abundant molecule on Earth, water. The process, called oxygenic photosynthesis, released a waste gas that nobody had ever seen in such quantities before. That waste was oxygen, and at first it seemed harmless enough. These tiny innovators, known today as cyanobacteria, began multiplying in the sunlit shallows. They formed vast green and blue mats that stretched across the coastal waters, feeding on carbon dioxide and pouring out oxygen as a byproduct. For the first time in Earth history, a living organism was producing free oxygen in massive amounts. And they did not stop. Generation after generation, they kept splitting water and releasing oxygen into the ancient seas. The planet's natural buffers, the iron dissolved in the oceans, could only absorb so much. As the cyanobacteria spread, the rust they created settled to the seafloor in banded layers, evidence we can still see today, but eventually the iron ran out. The oceans could no longer soak up the waste. Oxygen began to build up in the water and then bubble into the atmosphere. The cyanobacteria themselves were protected by their own internal chemistry, but for every other living thing on Earth, this was the beginning of a slow-motion catastrophe. They were releasing a poison into the very air the world breathed, and they had no idea what they were doing. Inside each cyanobacterium, a tiny revolution was taking place. Deep within its cell, a protein complex called Photosystem II was doing something that no other life form on Earth had ever attempted. It was using the energy from sunlight to tear apart a water molecule. That simple act, splitting H2O into hydrogen and oxygen, sounds almost mundane to us now, but around 2.7 billion years ago, it was a biological breakthrough that would change the entire planet. The hydrogen was used to build sugars and fuel the microbes' growth. The oxygen, however, was simply discarded, a waste product that floated away into the surrounding water. At first, this waste did not travel far. The early oceans were still rich in dissolved iron, and that iron eagerly grabbed any free oxygen it encountered, turning into rust and sinking to the seafloor. So the oxygen released by these early cyanobacteria was trapped locally, creating small pockets where the gas could accumulate only if no iron or other reactive elements were nearby. These pockets became known to scientists as oxygen oases, tiny islands of oxygenation in a vast anoxic world. Around each thriving colony of cyanobacteria, the water chemistry began to shift. The levels of free oxygen rose ever so slightly, just enough to create a chemical boundary. Beyond the edge of an oasis, the global ocean remained as it had always been, dark, reducing, and deadly to anything that depended on oxygen. But inside that oasis, a new kind of environment was emerging. It was a fragile and temporary haven, a proof of concept that oxygen could exist in the wild. For the anaerobic microbes that had ruled the seas for so long, these oases were toxic zones. Venturing too close meant oxidative damage and death, so the cyanobacteria continued their slow spread, dotting the sunlit shallows with their microscopic factories. They did not migrate or plan. They simply grew where the light was good, releasing oxygen as a byproduct of their own survival. And with each new oasis, the ancient chemical balance of the world tipped just a little further toward a future no one could have predicted. Oxygen's toxicity came from its hunger for electrons. Inside the cells of ancient anaerobes, every molecule was carefully built to function without this reactive gas. When oxygen appeared, it immediately began snatching electrons from the delicate machinery of those cells. It oxidized their membranes, their proteins, and even their DNA. This process is similar to what happens when iron rusts or when a sliced apple turns brown. The oxygen strips away electrons, causing the structure to break down and lose its function. For an anaerobic microbe, this was catastrophic. They had no defenses against it. Their entire biology was like a wooden house left out in a rain of sparks. But the real danger came from a particularly aggressive form of oxygen called reactive oxygen species. These are not the stable O2 we breathe today. They are unstable, charged fragments that form when oxygen interacts with other molecules inside a cell. Think of them as tiny, volatile sparks that bounce around and set off chain reactions. A single reactive oxygen molecule can damage dozens of nearby molecules before it is neutralized. It tears apart the fatty acids that make up cell membranes, pokes holes in the protective outer layer, and corrupts the genetic instructions stored in DNA. For early life, this was like a molecular wildfire spreading unchecked through the cell. The most common of these dangerous fragments is the superoxide radical. It forms when oxygen gains an extra electron. That extra electron makes it frantic and unstable. It will immediately try to offload that energy by reacting with anything it touches. Another is hydrogen peroxide, which sounds familiar from household cleaning bottles. Inside a cell, hydrogen peroxide can break apart into hydroxyl radicals, which are even more aggressive. Hydroxyl radicals will attack almost any organic molecule they encounter, pulling electrons away and leaving behind a trail of broken bonds. These reactive species are what scientists call oxidative stress. And for organisms that had never faced such a threat, the arrival of oxygen was like a constant chemical assault from within. The anaerobes had no antioxidants, no enzymes to neutralize these sparks. They had no repair systems to fix damaged DNA or patch up ruptured membranes. So when oxygen levels began to rise, even the small amounts near those early oases, entire populations of microbes were wiped out. The very gas that would one day power complex life was at that moment a merciless poison. It did not discriminate. It attacked the smallest and most vulnerable cells with equal ferocity. And yet, as we will see, some microbes had a secret. They carried a faint flicker of protection, a tiny defense that would allow them to survive this chemical war and eventually become the ancestors of every animal that ever lived. Inside those defenseless cells, the damage unfolded like a chain reaction of tiny explosions. Each oxygen molecule that drifted in became a threat, but the real danger came from the unstable fragments it spawned. These fragments, known as reactive oxygen species, included superoxide and hydrogen peroxide. Superoxide formed when oxygen grabbed an extra electron, leaving it frantic and hungry for stability. It would immediately lash out, stealing electrons from whatever it touched, like a spark landing on dry grass. Hydrogen peroxide was quieter but just as destructive. It could slip through membranes and then break apart into hydroxyl radicals, the most aggressive of all. Hydroxyl radicals attacked DNA, ripping apart the genetic instruction manual. They tore into proteins, unraveling their shape and destroying their function. They punched holes in cell membranes, turning the protective outer layer into a sieve. For early anaerobes, this was like a fire spreading through a wooden house with no fire extinguisher anywhere in sight. They had no antioxidant enzymes, no defenses to neutralize these sparks. They could not repair the damage because the damage happened faster than any repair could work. Even trace amounts of oxygen, concentrations far below what we breathe today, caused cellular chaos. A few molecules of oxygen could poison an entire colony of microbes. It was a chemical war fought on a microscopic scale, and the anaerobes were losing. But somewhere in that chaos, a few survivors carried a hidden advantage. They had a faint, ancient ability to recognize oxygen and render it harmless. That ability would become the seed of a new world. But the quiet resistance of a few microbes was not enough to stop the rising tide. The oxygen kept building, and the world entered a crisis that scientists call the Great Oxidation Event. It was not a sudden explosion, but a slow, relentless poisoning that unfolded over hundreds of millions of years. The cyanobacteria kept splitting water, and the oxygen they released began to accumulate in the oceans and then leak into the atmosphere. By around 2.4 billion years ago, the planetary buffers had finally been overwhelmed. The iron in the seas had turned to rust and sunk to the bottom. The volcanic gases that once soaked up free oxygen had been consumed. Now there was nothing left to stop this toxic gas from spreading. It rose from the water and mixed with the air, creeping across the entire planet like a silent fog. For the vast majority of life, this was catastrophic. The anaerobes that had dominated the Earth for nearly 2 billion years had no defense against even trace amounts of oxygen. Their cells began to break down on a global scale. Entire ecosystems collapsed. the shallow seas that once teemed with microbial mats turned into chemical wastelands where the very element that had become abundant was now a killer. This was not a localized disaster. It was a planetary extinction event, one of the most severe in Earth's history. Estimates suggest that somewhere between 90 and 99% of all living things may have been wiped out during this period. The survivors were those that either found refuge in oxygen-free pockets or carried the faint seeds of resistance that we mentioned earlier. The world itself changed too. The atmosphere transformed from a reducing haze into an oxidizing one. Iron that had once been dissolved in the oceans was now locked away in rust-colored rock formations. The sky began to turn blue because ozone, a form of oxygen, started to form a protective layer high above. But that beauty came at a terrible cost. The Great Oxidation Event was a planetary poisoning, a slow death for an entire world of life that had evolved in the absence of oxygen. It was the first and perhaps the greatest of the mass extinctions, and it cleared the stage for a new kind of biology to emerge. The microbes that survived would carry the tools to manage oxygen, and those tools would become the foundation for everything to come. The buffers could only hold for so long. For hundreds of millions of years, Earth had managed to soak up the oxygen that cyanobacteria released. Dissolved iron in the oceans grabbed it eagerly, turning to rust and settling to the seafloor. Volcanic gases like hydrogen and methane reacted with it, neutralizing the threat before it could build up. But those buffers were not infinite. The iron eventually ran out, locked away in banded rock formations that we can still see today. The volcanoes could not keep pace with the relentless output. Meanwhile, the cyanobacteria kept multiplying, kept splitting water, kept pouring out their waste. By around 2.4 billion years ago, the planetary sponge was full, and then everything changed at once. The oxygen that had been trapped in the oceans and absorbed by minerals suddenly had nowhere to go. It began to bubble out of the water and flood the atmosphere. This was not a gradual trickle anymore. It was a rapid, global surge, a chemical tipping point that scientists call the Great Oxidation Event. In a geologically short span of time, the concentration of free oxygen in the air shot up from nearly zero to perhaps a few % of modern levels. That may not sound like much, but for the life of that era, it was catastrophic. The vast majority of organisms had evolved in a world without oxygen. Their cells had no defenses against this reactive gas. They could not even tolerate a trace of it. As the oxygen spread across the planet, it poisoned the oceans and the air. Whole communities of microbes perished, their membranes torn apart, their DNA shattered. The extinction was staggering. Some estimates suggest that between 90 and 99% of all living things disappeared during this period. The blue sky that began to form above, thanks to a new ozone layer, came at the cost of an almost empty world. This was the first great die-off, and it reshaped the planet completely. The survivors were few, but they carried the seeds of a new era. The evidence of that planetary crisis remains locked in stone. Billions of years later, geologists can still read the story of oxygen poisoning in layers of ancient rock called banded iron formations. These striking deposits, found on every continent, consist of thin alternating bands of rust-red iron oxide and pale gray chert, a type of quartz. They look like the pages of a book, each one recording a tiny chapter of Earth's chemical history. And they tell a remarkable tale about how oxygen first began to poison the oceans. The process started when cyanobacteria released oxygen into the water. That oxygen immediately reacted with the vast amounts of dissolved iron that had accumulated in the ancient seas. The reaction created iron oxide, which is essentially rust, and that rust was heavy. It sank slowly through the water column and settled onto the seafloor, forming a thin layer of sediment. Then, over time, the oxygen production would pause or slow, perhaps because of seasonal changes or shifts in the microbial population. During those quiet periods, silica and other minerals would settle instead, creating a layer of chert. Then the cyanobacteria would resume their work, another burst of oxygen, another layer of rust. This rhythmic cycle repeated itself for hundreds of millions of years, building up stacks of banded rock that could be hundreds of feet thick. Each band is a tiny snapshot of the war between oxygen and iron. The iron was acting as a buffer, a chemical sponge that soaked up the toxic gas before it could accumulate in the air. But as the formations grew thicker, they also recorded the slow exhaustion of that buffer. Eventually, the iron in the oceans ran out, the bands stopped forming, and oxygen was free to escape into the atmosphere. That is when the Great Oxidation Event truly began. Today, these banded iron formations are among the most valuable sources of iron ore on the planet. But to a geologist, they are something far more precious, a timeline written in stone showing exactly how oxygen transformed a world. They are the silent witnesses to a planetary poisoning, and they remind us that the air we breathe today was not always a gift. It was once a weapon, and these rocks hold the scars of that ancient battle. Those banded iron formations tell a remarkably clear story. Each layer of rust-red rock represents a chapter in Earth's chemical history, a record of oxygen reacting with dissolved iron in the ancient oceans. When cyanobacteria released their waste, the free oxygen immediately bound to iron atoms suspended in the water, forming iron oxide particles that sank slowly to the seafloor. Over millions of years, these particles accumulated in thin, alternating bands, creating the striped sedimentary rocks that geologists now find across every continent. The process was like a planetary filtration system, a natural sink that absorbed the toxic gas before it could build up in the atmosphere. But that sink had a finite capacity. As the oceans gradually exhausted their supply of dissolved iron, the bands stopped forming. The last of those iron-rich layers settled into place around 1.8 billion years ago, marking the moment when the buffer was finally full. From that point onward, oxygen had nowhere to go but up. It began to accumulate in the air slowly at first, then with increasing speed, triggering the Great Oxidation Event. These rocks are more than just a geological curiosity. They are a timeline, a precise marker that tells us exactly when Earth crossed the threshold from an anoxic world to one that would eventually support complex life. The disappearance of those iron bands is the signature of a planet transformed. When the planetary buffer finally gave out, the invisible tide of oxygen swept across the globe with devastating speed. Within a geologically short span, the gas that had been trapped in the oceans and absorbed by minerals now flooded the atmosphere and the shallow seas. For the ancient anaerobic microbes that had ruled Earth for nearly 2 billion years, there was nowhere to hide. Their cells had evolved in a world without free oxygen, and they possessed no defenses against its corrosive power. Every breath of this new gas was a chemical attack from the inside out. The die-off was not a sudden explosion, but a relentless, silent war waged on a microscopic scale. As oxygen molecules drifted into the cells of these organisms, they immediately began to strip electrons from essential proteins and fats. The cell membranes, once flexible and protective, grew brittle and leaky. The DNA, the very blueprint of life, was torn apart by reactive fragments that formed in the presence of oxygen. One by one, the microbial communities that had carpeted the seafloor for eons collapsed. The vibrant green and purple mats that once shimmered in the shallows turned pale and then vanished, replaced by layers of rust-colored sediment. The oceans became vast killing fields, where the very element that would one day sustain complex life was now a poison of unparalleled efficiency. Scientists estimate that between 90 and 99% of all living things on Earth perished during this period. It was the first great extinction, far more sweeping than the one that would later end the reign of the dinosaurs. The survivors were those that had either found refuge in oxygen-free pockets deep beneath the sediments, or carried the faint genetic tools to neutralize the threat. These few resilient microbes held the key to a new world, but for the vast majority, the age of anaerobic dominance was over. The planet had been reset, and a new, oxygenated chapter was about to begin. Yet, even as the old world was wiped away, a question hung in the air. What could possibly survive such a catastrophe, and how would it transform the future of life? The survivors were few, and they were scattered. As oxygen spread through the ancient seas and sky, the vast majority of anaerobic microbes had only one option: to flee. They retreated into the last places where the old world still existed, deep within seafloor sediments, inside oxygen-free basins, and around hydrothermal vents where volcanic gases still bubbled up, untouched by the new poison. These refugia became islands of the past, small pockets where life could continue as it had for billions of years. But the retreat was not orderly. It was a desperate, slow-motion scramble across geological time. Entire populations were wiped out before they could find shelter. The shallow seas, once teeming with microbial mats, became barren zones where nothing could survive. The oxygen did not attack all at once. It crept in, layer by layer, year by year, pushing the boundary of the habitable zone deeper and deeper. What had been a global ecosystem was now reduced to a patchwork of isolated sanctuaries. To understand the scale of this collapse, it helps to compare it to later mass extinctions. The one that ended the reign of the dinosaurs, for example, was sudden, triggered by an asteroid impact that changed the climate in a geological blink. But the Great Oxidation Event was different. It unfolded over tens of millions of years, a slow, relentless poisoning that gave life no single moment of crisis to rally against. It was like a rising tide, not a wave. The anaerobes that survived were those that happened to live in the right places, deep enough, anoxic enough to escape the creeping gas. They did not evolve new defenses in time, they simply hid. And when the oxygen levels finally stabilized, the world that emerged was almost empty. Estimates suggest that more than 90% of Earth's biodiversity had vanished. The planet was stripped down to a fraction of its former richness. The survivors clung to the edges of existence, in the darkness of the deep ocean, in the sediments beneath the seafloor, in the chemical vents where hydrogen sulfide still flowed. They became the quiet keepers of an ancient lineage, a biological legacy that would never again dominate the surface, but their story was far from over. While they waited in the shadows, another group of microbes was learning to do something extraordinary. Those that had weathered the poison were not just hiding. They were changing, and their transformation would set the stage for everything to come. The survivors huddled in their deep-sea sanctuaries, but the planet above them was plunging into an ice age unlike any before. The same oxygen that had poisoned the oceans now triggered a chain reaction that would freeze the entire surface of the Earth. To understand why, we have to look at what happened to methane, the powerful greenhouse gas that had kept the young planet warm. For billions of years, methane had filled the atmosphere, produced by volcanic eruptions and the metabolic activity of anaerobic microbes. It trapped heat from the sun like a thick blanket, maintaining a global temperature that allowed liquid water to exist despite a dimmer sun. But oxygen changed everything. When methane molecules drifted into the oxygen-rich air, they reacted. Sunlight provided the spark, and the methane broke apart, turning into carbon dioxide and water vapor. Both were weaker greenhouse gases. The blanket began to thin. As oxygen levels rose, more methane was destroyed, and as methane disappeared, the planet started to cool. This created a feedback loop. More oxygen meant less methane meant colder temperatures, and colder temperatures meant less biological activity, which could have slowed oxygen production. But the cyanobacteria were still thriving in the sunlit shallows, pumping out oxygen despite the chill. The cooling continued, unchecked. The ice caps grew, creeping down from the poles toward the equator. The ocean surface began to freeze, and soon the entire planet was encased in ice, a state that scientists call Snowball Earth. The world became a frozen orb, reflecting more sunlight back into space and locking itself into a deep freeze that would last for millions of years. For the handful of microbes still clinging to life near volcanic vents and beneath the ice, this was the ultimate test. The oxygen-methane feedback loop had turned the Earth into a freezer, and the very gas that started it all was now threatening to finish the job entirely. The very gas that had kept the early Earth warm was being systematically erased. Methane, a powerful greenhouse gas produced by volcanoes and anaerobic microbes, had maintained a cozy blanket around the planet for billions of years. But now that blanket was unraveling. As oxygen levels climbed, it reacted with methane in the atmosphere, using sunlight as a catalyst to break the methane apart. The reaction turned each methane molecule into carbon dioxide and water vapor. And while carbon dioxide is also a greenhouse gas, it is far less effective at trapping heat than methane. Molecule for molecule, methane is about 25 times more potent. So as the methane disappeared, the blanket thinned dramatically. The planet began to lose heat faster than it could retain it. The cooling was not gradual. It accelerated as more oxygen poured in, destroying more methane, which allowed the planet to cool further, which slowed biological activity, but the cyanobacteria kept pumping out oxygen in the sunlit waters that remained. The feedback loop was vicious. Within a relatively short geological window, the global temperature dropped so sharply that ice began to form at the poles and creep toward the equator. The oceans froze over, the continents were buried under miles of ice. The entire surface of the Earth became a single, shimmering white orb, reflecting most of the sun's energy back into space and locking itself into a deep freeze. This was the Huronian glaciation, a Snowball Earth episode that would last for hundreds of millions of years. For the surviving anaerobes in their deep-sea refuges, the crisis had just become far worse. Not only did they have to hide from the poison of oxygen, but now their entire world was encased in ice. The shallow seas where cyanobacteria once thrived were sealed off from sunlight, cutting off the production of new oxygen. But the damage was already done. The atmosphere had already been transformed. The oxygen that remained would continue to react with any methane that tried to rebuild, keeping the planet frozen for eon after eon. The very element that would one day power complex life had turned the Earth into a frozen wasteland, a cruel irony that would test the limits of survival itself. And yet, beneath the ice, life persisted, waiting for the glaciers to finally retreat and for a new chapter to begin. Deep beneath the ice, a quiet revolution was unfolding inside the cells of a few lucky survivors. These microbes had not simply hidden from oxygen. They had begun to build shields against it. The first line of defense was a family of enzymes that scientists now call antioxidants, though the name makes them sound like something you would buy at a health food store. In reality, these were microscopic warriors, tiny proteins that could seek out and neutralize the reactive oxygen species before they could cause damage. The most important of these early defenders was an enzyme called superoxide dismutase. It targeted the superoxide radical, the most common and destructive of the oxygen fragments. Think of superoxide dismutase as a miniature fire extinguisher. It would grab hold of a superoxide molecule and break it apart into hydrogen peroxide and ordinary oxygen. That might not sound like a victory because hydrogen peroxide is still dangerous, but the cell was not finished. Another enzyme, catalase, could then split hydrogen peroxide into harmless water and oxygen. Together, these two enzymes formed a two-step detoxification system that could render the most aggressive oxygen byproducts completely safe. The evolution of these enzymes was not a sudden invention. It was a slow, piecemeal process that took millions of years. a random mutation would produce a protein that happened to have a weak ability to react with oxygen radicals. That tiny advantage was enough to help a cell survive a little longer, reproduce a little more. Over many generations, natural selection refined those crude proteins into efficient, specialized enzymes. The cells that carried them became more resistant to oxygen poisoning. They could venture closer to the oxygen-rich zones near the surface, where food was more abundant. They could outcompete their defenseless neighbors. This was the birth of aerobic metabolism, the ability to not just tolerate oxygen but to use it as a source of energy. The same enzymes that protected the cell from oxygen also allowed it to harness the gas for a far more powerful form of respiration. It was a double breakthrough: survive the poison, then turn it into fuel. The microbes that perfected this trick would become the ancestors of every oxygen-breathing creature on Earth, including us. Their small biochemical innovation, the evolution of antioxidant systems, was the key that unlocked the door to a new world. These molecular shields demanded constant energy and materials. Building and maintaining enzymes like superoxide dismutase and catalase was not free. For a tiny microbe living in a resource-limited world, every protein it made required precious amino acids and ATP, the energy currency of the cell. Investing in defenses meant less energy for growth and reproduction. It was a trade-off that slowed down the survivors, making them less competitive in the oxygen-free zones they had fled. But in the oxygen-rich regions that were expanding across the planet, that cost became an absolute necessity. Without these shields, any microbe that drifted into a pocket of oxygen would be destroyed within minutes. With them, a cell could tolerate low levels of the gas, enough to survive near the edges of the toxic frontier. This tolerance was not yet the ability to use oxygen for energy. It was more like living in a house built to withstand a constant drizzle of sparks. The roof might hold, but you still had to stay alert. The superoxide dismutase acted as a rapid response team, neutralizing the most aggressive radicals as soon as they formed. Catalase handled the hydrogen peroxide that slipped through. Another family of enzymes called peroxidases tackled additional reactive fragments, using electrons from other molecules to disarm them. Together, these three lines of defense formed a layered detoxification system. But even with all three, the cell was still fighting a losing battle if oxygen levels rose too high. The enzymes could only work so fast. If the concentration of oxygen surged, the reactive species would overwhelm the defenses, and the cell would die. So for hundreds of millions of years, these early aerobic microbes lived in a narrow band, just deep enough to avoid the worst of the poison but shallow enough to access sunlight or organic nutrients. They were pioneers of a new metabolic frontier, and their costly adaptations were the first fragile bridge between an anaerobic past and an oxygenated future. Each generation that survived passed down the genetic instructions for these shields. Over time, natural selection refined the enzymes, making them more efficient and less expensive to produce. The cost never disappeared, but it became manageable, and that management was the crucial first step. It allowed life to stop simply hiding from oxygen and start coexisting with it. That coexistence would eventually become cooperation, a partnership that would unlock vast new reservoirs of energy. But before that could happen, the world had to wait for the ice to melt, for the chemical balance to shift, and for a few bold microbes to take the next leap. But simply surviving the poison was only half the story. The same enzymes that protected those early microbes from oxygen damage also handed them the keys to a staggering new power source. Once a cell could neutralize the reactive fragments, it was only a small evolutionary step away from deliberately funneling oxygen into its energy-making machinery. This was the aerobic revolution, the moment when life stopped treating oxygen as a threat and began using it as fuel. the difference in energy yield between anaerobic and aerobic respiration is almost hard to wrap your mind around. An anaerobic microbe feeding on a simple sugar molecule through fermentation could extract about 2 molecules of ATP, the basic energy currency of the cell. That is enough to survive, but barely. A microbe that could use oxygen in a process called respiration could extract 36 or more ATP from the same sugar molecule. That is an 18-fold increase. It is the difference between getting around on foot and stepping into a rocket ship. The secret lay inside the cell's membrane, where a chain of proteins evolved to pass electrons down a series of steps like a bucket brigade. At the end of that chain sat oxygen, ready to accept the exhausted electrons. This final step, this pairing of oxygen with electrons, released a massive burst of energy that the cell could capture and store. The process became known as the electron transport chain, and it was the single most efficient energy harvesting system life had ever invented. The microbes that perfected this trick could grow faster, reproduce more, and outcompete their anaerobic neighbors in any environment where oxygen was present. They did not just survive the poison, they rode it to dominance. Their populations exploded, spreading through the shallows and then into the deeper waters where oxygen had begun to penetrate. For the first time, life had a metabolic engine that could power more than just simple, single-celled existence. The extra energy opened up possibilities that had never existed before. Cells could afford to grow larger, they could carry more DNA, they could build complex internal structures. The aerobic revolution did not just change how cells made energy. It changed what a cell could become, and it set the stage for the most dramatic transformation in the history of life, the leap from single cells to multicellular creatures. That extra energy changed everything about how cells could live, but the real breakthrough came when a few microbes stopped simply tolerating oxygen and began actively pulling it into their energy-making machinery. Instead of treating the gas as a toxin to be neutralized, they started using it as a final electron acceptor in a carefully controlled chain of reactions. This was the birth of aerobic respiration, and it rewrote the rules of survival. Inside the cell, a series of proteins evolved to pass electrons down a line, like a bucket brigade moving water toward a fire. At the very end of that line sat oxygen, waiting to accept the exhausted electrons. When oxygen grabbed them, it released a huge burst of energy that the cell could capture and store. The difference this made was staggering. A single molecule of glucose, when broken down through fermentation, the best that anaerobic life could manage, yielded just 2 molecules of ATP, the cell's main fuel. That is barely enough to keep a microbe alive and replicating, but the same glucose molecule, run through aerobic respiration, could produce 36 or more ATP. That is an 18-fold increase from the same starting material. It is like getting 18 days of work out of a single day's worth of food. This energy surplus was not a small advantage. It was a revolution that opened doorways that had been sealed for billions of years. With that much extra fuel, cells could grow larger. They could build more complex internal structures. They could carry bigger genomes and engage in more sophisticated behaviors. The energy budget of a single aerobic microbe was suddenly vast enough to support tasks that had been impossible for its anaerobic ancestors. But this transformation only became possible after oxygen had stopped being a universal poison. The same gas that had wiped out nearly all life had to be tamed first. The evolution of antioxidant defenses created a buffer, a window of tolerance, and within that window, a few bold microbes discovered that the very molecule that had nearly destroyed their world could be harnessed as a source of power. They did not just survive the poison, they learned to drink from the same fire that had burned their ancestors, and that discovery set the stage for the most profound shift in the history of life. Complex cells were no longer a distant dream, they were becoming an energetic possibility. The leap from simple microbes to complex cells required a partnership that changed the rules of life forever. Somewhere in the ancient oceans, a larger microbe swallowed a smaller one, but instead of digesting it, the two began to live together. The smaller one was an oxygen-breathing bacterium, and it found a safe home inside the larger cell. Over time, this internal guest became a permanent resident, eventually evolving into the organelle we now call the mitochondria. This was the birth of the eukaryote, a cell with a nucleus and a powerful internal engine. The new arrangement was a trade. The host provided shelter and nutrients, while the resident churned out vast amounts of energy from oxygen. That energy surplus allowed eukaryotes to grow 100 times larger than any bacterium. They could support a nucleus to store and protect their DNA. They could build internal membranes to compartmentalize different tasks. They could even begin to form colonies of specialized cells, the first stirrings of multicellular life. This fusion of two distinct organisms was not a gradual change. It was a sudden, irreversible merger that created a new kind of cell architecture. Without that partnership, the energy needed for complex bodies and brains would never have existed. The mitochondria became the power plants of every animal, plant, and fungus that would ever evolve, and they carried with them a reminder of their bacterial past, their own separate DNA, still coiled inside them like a tiny living fossil. So the next time you take a breath, remember that the oxygen you inhale is being fed to ancient bacterial guests living inside every one of your cells. They are the real reason you are here, the silent partners that turned a planetary poison into the fuel for a complex world. That ancient bacterial guest did not simply drift into its new home by luck. The event that created the first complex cell was a precise and improbable act of capture. According to the endosymbiotic theory, a larger anaerobic host cell, a member of a group called archaea, swallowed a smaller oxygen-breathing bacterium. Instead of digesting it, the host kept it alive, and over time the two began to cooperate. The host offered a protected environment, shelter from the harsh oxygen-filled world outside. The guest offered a powerful energy engine, the ability to extract far more fuel from the same food than any anaerobic cell could manage. This partnership was not a casual friendship. It was a merger of two very different survival strategies, one built on avoiding the poison and the other on harnessing it. The host still carried the old anaerobic protections, the enzymes that neutralized reactive oxygen fragments. The guest brought the machinery for aerobic respiration, the chain of proteins that turned oxygen into a torrent of energy. Inside the same cell, these two systems had to work together without destroying each other. The host had to keep its internal environment carefully balanced, ensuring that the oxygen delivered to the guest did not leak out and damage its own delicate structures. It was a constant act of chemical diplomacy. The host would package oxygen tightly, funneling it directly to the guest's membrane. Any stray oxygen molecules that escaped had to be quickly neutralized by the host's antioxidant enzymes. This delicate arrangement required the host to invest even more energy in maintaining those defenses, but the payoff was enormous. The guest could produce up to 36 ATP from a single sugar molecule, while the host alone could only manage 2. That surplus of energy allowed the host cell to grow larger, carry more DNA, and eventually evolve new structures like a nucleus. The guest itself began to change. Over millions of generations, it lost most of its own genes, becoming dependent on the host for its survival. It shrank down into a tiny organelle, wrapped in a double membrane, with its own separate DNA still coiled inside. This is the pattern we see in every complex cell today. Mitochondria are not just cellular parts. They are the descendants of free living bacteria, and they still carry the signature of their ancient independence. The endosymbiotic event was a gamble that paid off beyond any measure. It created the first eukaryote, the ancestor of every animal, plant, and fungus that would ever exist. And it required a constant, quiet vigilance over the very gas that had once nearly sterilized the planet. Inside that cell, oxygen was no longer a poison. It was a controlled resource, a fuel to be measured and delivered with precision. The partnership between host and guest became the foundation for all complex life, and it still hums inside your body right now, in every cell that burns oxygen to keep you alive. The energy surplus from oxygen did not just allow cells to grow bigger, it allowed them to rewrite their own genetic blueprints in ways that had never been possible before. A larger genome requires far more energy to replicate, repair, and express. Every time a cell copies its DNA, it burns through a significant amount of ATP. For anaerobic microbes, with their meager 2 ATP per sugar, the cost of carrying extra genetic material was a luxury they could rarely afford. Their genomes stayed small and streamlined, stripped down to the essentials for survival. But once mitochondria began churning out 36 ATP from the same meal, the energy budget expanded dramatically. Cells could suddenly support tens of thousands of genes instead of just a few hundred. This extra genomic real estate opened up space for innovation. Gene duplication became common, where a copy of an existing gene would appear by accident. Normally, a duplicate would be useless or even harmful, consuming resources without providing a benefit. But in an energy-rich cell, the duplicate could sit idle for millions of years, accumulating mutations without affecting the original function. Occasionally, one of those mutations would produce a new protein with a useful new job. Over time, these duplicates gave rise to entire families of enzymes, structural proteins, and signaling molecules that allowed cells to sense their environment, communicate with neighbors, and build internal frameworks. The genome itself became a laboratory for experimentation, and oxygen was the fuel that kept the lights on. This explosion of genetic complexity was not a sudden event. It unfolded over hundreds of millions of years, with each new gene adding a layer of possibility. Cells developed membrane-bound organelles, a nucleus to protect their DNA, and intricate cytoskeletons to maintain their shape. They could coordinate movements, engulf particles, and eventually form colonies where individual cells began to specialize. All of this was built on the foundation of aerobic respiration, the chemical partnership that turned a planetary poison into a source of boundless energy. The oxygen that once wiped out nearly all life had become the key that unlocked the genomic library of the living world, and that library would soon contain the instructions for something far more complex than any single cell could ever become. But that genomic library came with a steep price. Every new gene added to the cell's instruction manual increased the risk of damage from the very oxygen that paid for its expansion. The same reactive oxygen species that had once wiped out entire ecosystems now threatened the delicate machinery inside each eukaryotic cell. DNA, the long molecule that stored all those new genetic innovations, was especially vulnerable. When a reactive oxygen fragment struck a strand of DNA, it could break the backbone, scramble the sequence, or cause permanent mutations. For a simple bacterium with a small genome, the odds of a catastrophic hit were low, but the repair systems were also limited. For a eukaryote carrying tens of thousands of genes, the target area was vastly larger, and the stakes were far higher. A single mutation in a critical gene could cripple the cell or trigger uncontrolled growth. So along with the energy bonanza came a new burden, the need for constant surveillance and repair. Eukaryotes evolved elaborate antioxidant networks, not just to neutralize oxygen fragments before they struck, but also to patrol the genome and fix damage when it occurred. Enzymes like superoxide dismutase and catalase were joined by new classes of repair proteins that could snip out damaged sections of DNA and replace them with fresh copies. These systems consumed energy themselves, but the energy surplus from aerobic respiration made them affordable. It was a delicate balance. The more complex a cell became, the more it had to invest in protecting that complexity. Oxygen enabled the expansion of the genome, but it also constrained it by setting a limit on how much damage could be tolerated. A cell could only grow so large and carry so many genes before the rate of oxidative damage outstripped its capacity to repair. This trade-off shaped the evolution of all eukaryotes, from the first single-celled pioneers to the multicellular creatures that would eventually emerge. Complexity was never free. It was a loan taken out against the constant threat of oxygen's corrosive power, and every generation had to pay the interest in the form of relentless maintenance. That constraint is still with us today. Every cell in your body spends a significant fraction of its energy budget just fighting off oxidative damage. As you age, those repair systems gradually wear down and the damage accumulates. The very process that allowed life to become complex is also a fundamental driver of aging. Oxygen simultaneously enabled the rise of intricate biology and set the boundaries within which it could operate. It was both a gift and a leash. Oxygen did not rise in one smooth wave. The Great Oxidation unfolded in two distinct pulses, separated by more than 1 billion years of relative stability. That long pause between the first surge and the second is one of the most puzzling chapters in Earth's history, and it holds the key to understanding why complex life took so long to appear. The first pulse, around 2.4 billion years ago, was the dramatic poisoning event that wiped out nearly all anaerobic life. It pushed oxygen levels from near zero to perhaps 1 or 2% of modern levels. Then, for reasons that geologists are still debating, the rise stopped. The planet settled into a long, quiet middle age where oxygen concentration hovered at just a few percent, enough to fuel some microbes but far too low to support anything like an animal. What caused this billion-year pause? The answer seems to lie in the deep Earth itself. Over millions of years, tectonic activity slowly buried vast amounts of organic carbon and sulfur in the crust, locking away the reducing elements that could have soaked up oxygen. At the same time, the continents were growing, and their weathering added more nutrients to the oceans, feeding photosynthetic life that kept pumping out oxygen. But the planet's internal buffers still had enough capacity to keep the gas in check. It was a stalemate between production and consumption, and it lasted for over 1 billion years. Then, around 800 million years ago, something shifted. New, more efficient forms of photosynthesis evolved. The first green algae appeared, spreading through the sunlit seas and boosting oxygen production. Meanwhile, tectonic movements pulled continents apart, creating shallow seas where oxygen could accumulate. The second great pulse began. Oxygen levels climbed from a few percent to maybe 15 or 20% of modern levels, still low but now high enough to sustain the first simple animals. This two-step rise was not a steady march but a series of breakthroughs and plateaus, each one unlocking new possibilities for life. The first pulse cleared the old world and built the foundations. The second pulse provided the energy threshold that allowed creatures to move, hunt, and eventually think. Without that long pause, without the slow accumulation of geological changes that set the stage for the second rise, complex bodies would never have had the chance to evolve. The Great Oxidation left a legacy written in rock and in the oxygen we breathe. It was a slow, patient process that took nearly 2 billion years to complete the transformation. And when the second pulse finally arrived, it triggered the most spectacular explosion of life the planet had ever seen. But the planet did not lock in that early oxygen and hold it steady. For more than 1 billion years after the first great poisoning, oxygen levels wobbled up and down like a slow, uneven breath. They never climbed much above a few percent of modern concentrations, and sometimes they dropped even lower, nearly back to the anoxic baseline. This long oscillation kept Earth in a kind of chemical limbo, a world where enough oxygen existed to support a few resilient microbes, but not nearly enough to fuel anything with a moving body or a hungry mouth. The atmosphere and oceans were locked in a tug-of-war between producers and consumers. Cyanobacteria kept pumping out oxygen, but volcanic gases, buried organic matter, and deep-sea minerals kept soaking it up. Every time oxygen began to accumulate, some geological or biological process would pull it back down. The result was a frustrating plateau, a billion-year stretch where complex life remained an impossible dream. Then, around 600 million years ago, the balance finally tipped. A second great surge in oxygen began, one that would eventually push concentrations close to modern levels. Scientists call this the Neoproterozoic Oxygenation Event. What caused this second rise? A combination of forces working together. The supercontinent Rodinia was breaking apart, creating vast new shallow seas and coastlines where photosynthetic organisms could thrive. Glaciers that had covered the planet were retreating, releasing nutrients into the oceans and feeding algal blooms on an unprecedented scale. New, more efficient forms of phytoplankton evolved, capable of pumping out oxygen even faster than their ancient ancestors. At the same time, tectonic activity buried huge amounts of organic carbon deep in the Earth's crust, locking away the carbon that would have otherwise reacted with oxygen and removed it from the air. With less oxygen being consumed and more being produced, the gas began to accumulate in earnest. Over tens of millions of years, oxygen levels climbed from a few percent to perhaps 15 or 20%, then higher still, approaching the rich atmosphere we breathe today. This slow but steady rise set the stage for a biological revolution. For the first time, the oceans contained enough oxygen to support creatures that were more than just microscopic specks. Simple, soft-bodied animals began to appear in the fossil record, the strange and sometimes giant forms of the Ediacaran Biota. They were not yet mobile predators, but they were large, multicellular, and distinctly different from anything that had come before. They spread across the seafloor like living quilts, feeding on microbial mats and absorbing oxygen directly through their thin skins. Their existence was a direct result of the Neoproterozoic Oxygenation Event. Without that second surge, their bodies would have been starved for energy, unable to grow beyond a few cells. Oxygen was the fuel that made size possible. It was the chemical key that unlocked the door to a world where animals could move, sense, and eventually dominate. And just a few tens of millions of years later, that door would swing wide open in the most famous burst of evolutionary creativity the planet has ever known. The first creatures to walk through that door left behind a mysterious fossil record. They were the Ediacaran Biota, a collection of strange, soft-bodied organisms that appeared in the oceans around 575 million years ago. These were not the simple single cells or microbial mats that had dominated for billions of years. They were large, often flat, and shaped like fronds, discs, or quilts. Some grew as wide as a meter across, others were tiny, no bigger than a coin. They had no shells, no skeletons, no limbs, and no visible mouths. They were the earliest known experiments in multicellular life, and they were radically different from anything alive today. To understand them, you have to imagine a world without predators. There was nothing that could bite, tear, or chase. The Ediacaran seafloor was a quiet, slow-moving environment where these organisms simply sat, absorbing nutrients from the water or from the microbial mats beneath them. They did not need to move because movement would have wasted energy. With oxygen levels still low by modern standards, around 5 to 10% of today's concentration, every cell had to be efficient. Being large and flat helped because it maximized surface area, allowing oxygen to diffuse directly into their bodies without needing a circulatory system. They were like living solar panels, but instead of light, they harvested chemicals and dissolved organic matter. Their bodies were made of simple tissues, not the complex organs of later animals. Some paleontologists compare them to lichens or giant single cells, while others see them as failed experiments, creatures that explored a path that evolution later abandoned. They flourished for nearly 40 million years, slowly spreading across the shallow seas of the ancient supercontinent. Then, around 541 million years ago, they vanished. Their disappearance coincides with the beginning of the Cambrian period, when oxygen levels climbed higher and the first true animals with shells and legs and jaws appeared. It seems the Ediacaran Biota could not compete in a world that suddenly became faster and more dangerous. Their soft, stationary bodies were no match for burrowing scavengers or active predators. They were replaced by a new generation of life, one that moved, ate, and evolved at a pace the Earth had never seen. But those early experiments were not pointless. They proved that multicellular bodies were possible. They tested the limits of size and shape under low oxygen, and they set the stage for the explosion of diversity that was about to follow. The Ediacaran Biota were like the first rough sketches in a long creative process, imperfect but essential, and they left their imprint in the rocks as a reminder that every complex life form begins as a fragile experiment. Their bodies spread across the seafloor like living quilts stitched from nothing but thin layers of tissue. Some were shaped like ferns pressed flat against the sand. Others formed perfect concentric circles as if drawn by a careful hand. A few grew into long ribbed tubes that stretched a meter or more across the ancient seabed. These were the Ediacaran Biota, and they were unlike anything that had come before or anything that would follow. They were the first large experiments in multicellular life, and their strange forms were directly shaped by the oxygen levels of their world. The Ediacaran oceans held enough oxygen to support something larger than a single cell, but barely. Estimates suggest concentrations hovered around 5 to 10% of modern levels, enough to sustain thin, simple bodies, but not nearly enough to fuel muscles, organs, or active movement. So these creatures adapted by staying flat and wide. Every cell in their body had to be within reach of oxygen diffusing directly through their skin. There were no guts, no circulatory systems, no gills. They absorbed what they needed from the water surrounding them, and their entire body plan was a solution to that chemical constraint. A thicker body would have suffocated. A dense cluster of cells would have starved for oxygen at the center. So they spread out like living sheets, maximizing surface area while keeping every cell close to the outside world. But oxygen did not stay steady during the Ediacaran period. It fluctuated, sometimes rising gently, sometimes dropping back toward dangerously low levels. These swings left their mark on the fossil record. When oxygen was higher, some Ediacaran forms grew larger and more elaborate, developing branching fronds or quilted segments that increased their surface area even further. When oxygen dipped, the fossils show smaller, simpler shapes, as if the creatures shrank back to a more survivable size. It was a constant negotiation between the energy available and the body they could afford to build. A few species even developed holdfasts, root-like structures that anchored them to the seafloor, probably to reach slightly higher water where oxygen might be more abundant. Despite their size, these creatures remained astonishingly fragile. They had no hard parts, no shells, no skeletons, no way to defend themselves against damage. A single storm or shift in current could tear a frond apart. A drop in oxygen could cause whole populations to vanish from the fossil record for millions of years. Their complexity was real, but it was also precarious, balanced on a knife edge between enough oxygen and not enough. They were like early sketches drawn in sand, beautiful but easily erased. Then, around 541 million years ago, the balance shifted for good. Oxygen levels climbed higher and stayed higher, and the Ediacaran Biota, so perfectly adapted to their low and variable world, found themselves outmatched by a new kind of creature, one that could move, burrow, and consume. The fragile quilts of the Ediacaran were replaced by shells and legs and jaws. But before that happened, they proved something essential, that complex bodies could exist at all, even under tight constraints. They were the first proof that life could reach beyond the microscopic, and their strange, flat shapes remain in the rocks as a reminder that every step toward complexity is also a step into vulnerability. The moment oxygen crossed a threshold, evolution shifted into a new gear. Around 541 million years ago, the oceans changed in ways that would never be reversed. Oxygen concentrations climbed past a critical point, perhaps reaching 15% of modern levels or higher. That was enough to fuel something the Earth had never seen: active, mobile, hungry bodies. The Cambrian explosion was not a single event but a burst of biological innovation that unfolded over about 20 million years, a geological blink. Creatures that had once drifted or stuck to the seafloor now began to move with purpose. They burrowed into sediment, they swam through the water column. And for the first time, they hunted one another. Two forces drove this transformation. The first was oxygen itself. With more of the gas available, animals could sustain muscles that worked in bursts, a heart that pumped blood, and a nervous system that processed information quickly. A sluggish worm could suddenly contract its body to shoot through mud. A swimming arthropod could flick its tail to escape a predator. Every movement required energy, and oxygen provided that energy in abundance. The second force was the evolutionary arms race that oxygen kicked off. When one animal evolved a shell to protect itself, another evolved a claw to break that shell. When a predator became faster, its prey became faster too. Each adaptation triggered a counter-adaptation, and the cycle accelerated with every generation. The fossil record of this period is a window into that arms race. The first animals with hard skeletons appear in the earliest Cambrian layers. These were tiny tubes and caps made of calcium carbonate, simple shields against an increasingly dangerous world. Then came the trilobites with their segmented exoskeletons and compound eyes. They could roll into a ball for protection or scuttle across the seafloor to scavenge. Not long after, the first large predators emerged, creatures like Anomalocaris, a meter-long swimmer with grasping appendages and a circular mouth lined with sharp plates. It could chase down soft-bodied prey in a way that nothing before it had ever done. The size of these animals was directly linked to oxygen. Earlier, thin, flat bodies were the only option because diffusion limited how deep oxygen could reach. But with more oxygen in the water, animals could develop thicker tissues, internal organs, and even simple circulatory systems that carried oxygen to every part of the body. The result was a dramatic increase in maximum body size. Some Cambrian animals grew as large as a meter long, while most Ediacaran forms had been measured in centimeters. The speed of this transformation astonished the first paleontologists who uncovered it. They called it an explosion because in the rock record, complex animal fossils appear suddenly without obvious precursors. But we now understand that the groundwork was laid over billions of years. The microbial invention of photosynthesis, the slow rise of oxygen, the evolution of antioxidant defenses, the endosymbiotic merger that created complex cells, and the long plateau of low oxygen that tested every possible body plan. The Cambrian explosion was the moment when all those earlier steps finally paid off. Oxygen was no longer a constraint. It was a resource that could be spent freely. And life spent it on a furious burst of invention, creating the basic body plans that would shape animal evolution for the next half-billion years. Every major group of animals today, from arthropods to mollusks to chordates, the lineage that would eventually include vertebrates, first appears in the Cambrian fossil record. We are living with the legacy of that ancient arms race, still shaped by the oxygen that set it in motion. That final rise in oxygen at the Precambrian-Cambrian boundary was the chemical threshold that made the animal world possible. For billions of years, oxygen had hovered at levels that could only support thin, stationary bodies. Then, around 541 million years ago, the gas surged past a critical point, perhaps reaching 15% of modern atmospheric concentration or even higher. This was not a slow drift. It was a genuine shift driven by a combination of geological and biological forces, the breakup of the supercontinent Rodinia, the evolution of more efficient phytoplankton, and the deep burial of organic carbon that locked away reactive elements. The oceans suddenly held enough dissolved oxygen to sustain something the Earth had never seen, a creature with muscles, a gut, and a nervous system that could send signals fast enough to coordinate a strike or a escape. The Ediacaran Biota had been content to soak up oxygen passively. They stayed flat and wide because their cells could only reach a few millimeters from the surface. But the new oxygen levels allowed animals to build thicker bodies, internal organs, and even simple hearts that could pump blood rich in oxygen to every tissue. That meant a creature could grow larger without suffocating at its core. It could also move with purpose because muscles require a steady supply of oxygen to contract. And once movement became possible, so did active predation. The first predators did not need to be fast or especially fierce. They simply needed to be faster than their prey, and the extra oxygen gave them that advantage. In the fossil layers of the early Cambrian, we see the first traces of burrows, of animals tunneling through sediment to scavenge or escape. We see the first hard parts, tiny shells and spines that suggest a world where defense had become necessary. And we see the first large hunters like Anomalocaris, a meter-long swimmer with compound eyes and grasping appendages that could snatch soft-bodied prey from the water. This burst of diversity is often called an explosion, and it transformed the oceans in less than 20 million years. Almost every major animal group that exists today, from arthropods to mollusks to the chordates that would eventually give rise to vertebrates, first appears in the Cambrian fossil record. But it is important to understand that oxygen was a prerequisite, not the sole cause. The Cambrian explosion also required ecological triggers, the evolution of eyes, the rise of burrowing, the spread of hard skeletons that created new niches. Without enough oxygen, however, none of those innovations could have been sustained. A trilobite with a thick exoskeleton would have been impossible in the low-oxygen world of the Ediacaran because its muscles and nervous system would have starved for fuel. Oxygen was the chemical key that unlocked the door, and once it turned, the arms race of predator and prey, of speed and armor, of vision and locomotion began in earnest. The animal body became a machine for capturing energy, and that energy came from the very gas that had once poisoned the planet. The same oxygen that had wiped out nearly all life 2 billion years earlier was now feeding the most creative burst of evolution the Earth had ever known. It was a gift born from catastrophe, and it set the stage for everything that followed. That ancient oxygen war is still being fought inside your body right now, every second of every day. Every cell in your body is a carefully controlled oxygen-burning engine, and that engine requires constant maintenance to keep from tearing itself apart. The same reactive oxygen species that once devastated ancient microbes are still produced inside your mitochondria as a natural byproduct of respiration. Your cells have inherited the same antioxidant defenses that evolved billions of years ago, superoxide dismutase and catalase, still patrolling your tissues, still neutralizing those volatile fragments before they can cause chaos. But the balance is delicate, too much oxygen or too little, and the system starts to break down. Consider what happens when you exercise. Your muscles demand more energy, so your mitochondria burn oxygen faster. That increased burn generates more reactive species, and your body responds by ramping up its antioxidant production. Over time, regular exercise actually strengthens this defense system, making your cells more resilient. It is a form of training that reaches all the way back to the first microbes that learned to handle the poison. On the other hand, chronic stress, poor diet, or pollution can overwhelm your natural defenses, leading to a state scientists call oxidative stress. This is not a vague concept. It is a measurable imbalance where the production of reactive oxygen outpaces the body's ability to neutralize them. Over years, that imbalance contributes to aging, to tissue damage, and to many of the diseases that come with getting older. Your body also has to manage oxygen distribution with remarkable precision. Too little oxygen and your cells suffocate. Too much, and they suffer from a condition called hyperoxia, which can damage the lungs and other tissues. That is why premature babies, whose lungs are not fully developed, are given carefully monitored oxygen levels in incubators. It is also why deep-sea divers breathe special gas mixtures, because pure oxygen at high pressure becomes toxic. The same gas that sustains us can still poison us if the concentration or the context changes. Our entire physiology is built around this ancient compromise. We need oxygen to live, but we must constantly protect ourselves from its corrosive side. Your mitochondria are the direct descendants of that ancient bacterial guest that was swallowed 2 billion years ago. They still carry their own DNA, separate from the rest of your genome, and they still produce the vast majority of your cell's energy. But they also produce the majority of the reactive oxygen species that your antioxidant defenses have to handle. It is a relationship that began as a desperate partnership in a poisoned world, and it has never stopped being a negotiation. Every time you take a breath, you are feeding those tiny internal engines, and every breath also exacts a tiny cost in oxidative wear and tear. This is why we age, why our cells slowly accumulate damage over a lifetime, and why the rate of that damage is influenced by everything from our diet to our activity level to the air we breathe. But there is another layer to this story. Our bodies have also evolved sophisticated sensors that detect oxygen levels and trigger appropriate responses. When you ascend a mountain and the air thins, a protein called HIF, or hypoxia-inducible factor, kicks into action. It signals your kidneys to produce more red blood cells, which increases the oxygen-carrying capacity of your blood. It also triggers the growth of new blood vessels and shifts your metabolism to be more efficient. This is the same system that helped our ancestors survive ice ages and high-altitude migrations, and it is a direct legacy of the billion-year struggle between life and oxygen. You are walking around with a finely tuned oxygen management system that was forged in the fires of the Great Oxidation Event and refined through every subsequent extinction and expansion. The next time you feel your heart beat faster after a sprint, or notice your breathing deepen on a hike, remember that you are experiencing the living echo of an ancient catastrophe. Your body is not just a machine that burns oxygen. It is a treaty, a long-standing truce between a once deadly gas and the life that learned to master it. And that truce is renegotiated in every cell, with every breath, every single day. Your body runs this constant chemical negotiation with every heartbeat. Blood vessels thread through every tissue, delivering oxygen that must be released at just the right pressure and picked up again before it can cause harm. The molecule that makes this possible is hemoglobin, a protein packed inside your red blood cells that grabs oxygen in the lungs and carries it to the rest of your body. But hemoglobin does not simply hold onto oxygen like a tight fist. It has a clever binding curve that adjusts its grip depending on the surroundings. In the lungs, where oxygen is abundant, hemoglobin loads up eagerly. In the tissues where oxygen is scarce, it relaxes its hold and lets the gas diffuse out precisely where it is needed. This elegant system evolved over hundreds of millions of years, and its fine-tuning is a direct response to the ancient threat of oxygen toxicity. If hemoglobin held onto oxygen too tightly, cells would starve. If it let go too easily, oxygen would be released in the lungs instead of reaching the tissues. The curve sits in a perfect middle ground, a legacy of countless genetic adjustments that allowed our ancestors to survive in a world where oxygen levels fluctuated wildly. But even with hemoglobin's careful delivery, every cell in your body still faces the same danger that the first oxygen breathers faced. Inside your mitochondria, where oxygen is burned for energy, a small fraction of that oxygen inevitably escapes the controlled reaction and turns into reactive fragments. These are the superoxide radicals and hydrogen peroxide molecules that your ancient microbial ancestors first learned to neutralize. Your cells still rely on the same basic enzymes, superoxide dismutase and catalase, to disarm these threats. The difference is scale. Your body contains trillions of mitochondria, each one producing energy and each one leaking a tiny amount of reactive oxygen. Over a lifetime, that leak adds up to an immense oxidative load, and your antioxidant systems must work constantly to keep the damage under control. This is why you need nutrients like vitamin C and vitamin E, which act as backup antioxidants, donating electrons to quench reactive species before they can attack your DNA or cell membranes. Your body also has sophisticated sensors that monitor oxygen levels in real time. When oxygen drops, as it does when you climb a mountain or hold your breath, a protein called HIF, or hypoxia-inducible factor, activates a cascade of responses. It tells your kidneys to produce more red blood cells, boosting your blood's oxygen-carrying capacity. It also shifts your metabolism to be more efficient, reducing the amount of oxygen each cell needs. This system is a direct inheritance from the long period when Earth's oxygen was low and variable. It allowed our ancestors to survive glacial advances and high-altitude migrations, and it still protects you today. On the flip side, when oxygen levels rise too high, as happens when you breathe pure oxygen in a medical setting, your cells must ramp up their antioxidant production to avoid oxidative damage. That is why divers use carefully blended gas mixtures and why premature babies receive oxygen at precisely controlled concentrations. The same gas that sustains us can still become toxic if the balance tips too far. Every time you exhale, you are releasing the spent remains of this careful process. Your red blood cells, after dropping off their oxygen, pick up carbon dioxide and carry it back to the lungs. But along the way, some of that oxygen escaped into the surrounding fluid, and your antioxidant enzymes had to catch it. This cycle runs billions of times a day inside your body, a silent, tireless operation that began with a few lucky microbes learning to handle a poison. The next time you feel your pulse in your wrist, remember that each beat is pushing hemoglobin through miles of vessels, delivering a gas that once nearly sterilized the planet. And that gas is still dangerous, still corrosive, still demanding constant vigilance. Your body is a living monument to that ancient compromise, a delicate balance between energy and destruction, maintained by molecules that first appeared in a world of rust and ice. But there is a limit to how much oxygen life can handle, and that limit is just as real as the lower boundary. For every creature that depends on oxygen, there exists a Goldilocks zone, a narrow band where the gas is abundant enough to fuel activity, but not so concentrated that it becomes a poison. Push past that upper limit, and the same molecule that powers your muscles begins to damage them, overwhelming the antioxidant defenses that your cells have maintained for billions of years. This is the hidden danger of too much oxygen, and it is a constraint that has shaped the evolution of every breathing animal, including us. Consider what happens inside a cell when oxygen levels climb too high. The delicate balance between production and neutralization of reactive species tips. More oxygen means more opportunity for those volatile fragments to form, and the enzymes that normally catch them start to fall behind. It is like a fire extinguisher system designed for a small kitchen fire suddenly facing a blazing warehouse. The reactive species begin to attack the very structures they once helped build. Cell membranes become leaky, proteins lose their shape, DNA accumulates breaks. The cell is not being poisoned from the outside, it is being poisoned from within by the very fuel it depends on. This phenomenon is not just a laboratory curiosity. It has real, observable effects on living organisms. For example, premature babies born with underdeveloped lungs are often given supplemental oxygen to help them breathe. But that extra oxygen must be carefully monitored. If the concentration is too high, it can damage the delicate tissues of the retina, leading to a condition called retinopathy of prematurity. The same oxygen that saves their lives can also steal their sight if it is not delivered in precise measure. It is a stark reminder that oxygen, for all its benefits, is still a corrosive gas, and our bodies are only adapted to a specific range of concentrations. The same principle applies to deep-sea divers. At depth, the pressure increases, and so does the partial pressure of oxygen in the breathing gas. Below about 50 meters, breathing ordinary compressed air becomes dangerous. The oxygen molecules are forced into the bloodstream and tissues at such high pressure that they overwhelm the body's defenses. A diver can experience oxygen toxicity, which feels like a sudden onset of muscle twitching, tunnel vision, ringing in the ears, and eventually seizures. This is not a lack of oxygen. It is an excess, a flood that the antioxidant systems cannot contain. That is why deep divers switch to special gas mixtures, replacing some of the oxygen with helium or nitrogen to stay within that Goldilocks zone. The danger of too much oxygen is not limited to extreme medical or diving scenarios. It also plays a role in how animals have evolved over millions of years. During the Carboniferous period, around 300 million years ago, atmospheric oxygen levels peaked at nearly 35%, far higher than today's 21%. At that time, insects grew to enormous sizes. There were dragonflies with wingspans of 2 feet and millipedes as long as a person. Their growth was limited by the way oxygen diffuses through their tracheal tubes, and more oxygen allowed larger bodies. But that same abundance came with a hidden cost. Even slight fluctuations in oxygen levels could push those giant insects into the toxic zone, and when fire became more frequent in that oxygen-rich atmosphere, entire forests could burn with unmatched intensity. The Carboniferous period ended not because of too little oxygen, but because of too much, combined with a cooling climate that sealed the fate of those oversized creatures. We see the same tension in the history of the Earth's atmosphere itself. After the second great surge of oxygen during the Neoproterozoic, levels continued to oscillate. There were times when oxygen climbed high enough to fuel the evolution of large, active animals, but also times when it surged too high, causing widespread oxidative stress and even extinction events. The fossil record shows that periods of very high oxygen often coincide with bursts of evolutionary innovation, but they also correlate with extinctions, as if the planet itself was testing the limits of life's tolerance. Our own bodies bear the scars of this balance. The reason we age, the reason our tissues slowly accumulate damage over a lifetime, is partly because our antioxidant defenses cannot keep up with the steady leakage of reactive oxygen from our mitochondria. The very process that gives us energy also causes a slow, cumulative wear and tear. We are constantly walking a tightrope between enough oxygen and too much. The trick is that the line between enough and too much is not fixed. It changes with activity, with altitude, with the health of our cells. It is a dynamic boundary that life has been negotiating for over 2 billion years. So oxygen is not simply a friend or a foe. It is a chemical partner that demands constant management. Too little and you suffocate, too much and you burn from the inside out. The Goldilocks zone is the narrow window where energy production is high enough to sustain complex life, but oxidative damage remains low enough to be repaired. It is a delicate equilibrium that has allowed animals to evolve, to move, to think, and to build civilizations. And it is a reminder that even the most essential substances on Earth have a dangerous edge. The next time you take a deep breath, consider that you are filling your lungs with a gas that could just as easily destroy you as sustain you. The only thing keeping you in the safe zone is 1 billion years of evolution and a set of enzymes that never stop working. This narrow Goldilocks zone has held steady for hundreds of millions of years. Geologists can see it in the layers of ancient rock where the chemistry of the atmosphere left its signature. One of the most dramatic examples comes from the end of the Permian period, around 252 million years ago, when Earth experienced its most severe mass extinction. The cause is still debated, but the evidence points to a combination of volcanic eruptions and a collapse in oxygen levels. The oceans became anoxic, meaning they lost nearly all dissolved oxygen, and the land experienced a sharp drop in atmospheric oxygen. Creatures that had thrived for millions of years suddenly suffocated. The fossil record shows that entire ecosystems vanished in a geological blink, but what is less often discussed is what happened after that low point. In the recovery that followed, oxygen did not simply bounce back to its previous level. It overshot, climbing to concentrations that may have been even higher than today. For a brief period, the atmosphere became hyperoxic, saturated with the very gas that had been so scarce during the extinction. This surge should have been a boon for life, more energy, larger bodies, faster metabolisms. But instead, it may have caused its own wave of harm. Laboratory experiments on modern animals show that exposure to hyperoxic conditions triggers massive oxidative stress. Cells cannot keep up with the flood of reactive oxygen species. Tissues become inflamed, DNA breaks, and organisms age faster or die. The Permian-Triassic rebound was not a smooth return to balance. It was a violent oscillation, a swing from suffocation to poisoning, and it took millions of years for the system to stabilize. That oscillation is a clear sign that Earth's oxygen level is not determined by chance. It is regulated by a complex web of geological and biological feedbacks that keep the gas within a specific range. Too low, and animals cannot extract enough energy to move or grow. Too high, and the oxidative damage becomes lethal. The planet has been locked into this delicate dance for over 500 million years, ever since the Cambrian explosion proved that complex life was possible only within that narrow corridor. Every breath you take is a product of that ancient balancing act. The volcanoes that pump out reducing gases, the weathering of rocks that consume oxygen, the forests that produce it, and the deep sea sediments that bury carbon, all of these forces work together to keep the atmosphere in a habitable range. It is a self-correcting system, but it is not perfect. When something disrupts it, as happened during the Permian, the consequences are catastrophic. That is why the story of oxygen is not just a tale of poison turned medicine. It is a reminder that the conditions that allow complex life to exist are rare and fragile. We are living in a fortunate moment, a window of stability that has been open for just a fraction of Earth's history. And that window exists precisely because oxygen is both a gift and a threat, a molecule that can build or destroy depending on its concentration. The next time you watch a sunset, consider that the blue sky above you is a sign of that precise balance. The oxygen in the air is just enough to keep you alive, but not so much that it burns you from the inside. That balance took billions of years to achieve, and it is maintained by forces we are only beginning to understand. Yet the exact forces that triggered oxygen's rise remain surprisingly mysterious. Scientists have identified the broad strokes: the appearance of cyanobacteria, the exhaustion of geological buffers, the long plateau, and the second surge. But when you zoom in on the precise timing and the specific triggers, the picture blurs into a tangle of open questions. Why did the Great Oxidation Event begin when it did, around 2.4 billion years ago, and not hundreds of millions of years earlier or later? The cyanobacteria that produced oxygen had evolved well before that tipping point. They were already splitting water and releasing waste, so what finally pushed the planet past its chemical threshold at that particular moment? One possibility lies deep beneath the surface, in the slow churn of tectonic plates. The Earth's crust is constantly being recycled, with new rock forming at mid-ocean ridges and old rock sinking back into the mantle. Over time, this process changes the composition of the continents and the chemistry of the oceans. Some researchers suggest that a major shift in volcanic activity around 2.5 billion years ago reduced the amount of reducing gases being pumped into the atmosphere. With fewer hydrogen and sulfur compounds to react with oxygen, the gas could finally accumulate, but the timing of that shift is not easy to pin down. The rock record from such ancient eras is fragmentary, eroded, and buried. Another question centers on the evolution of the cyanobacteria themselves. Did they suddenly become more efficient at producing oxygen, or did they simply multiply to a critical population size? The fossil evidence is frustratingly sparse. Ancient microbial mats can leave behind structures called stromatolites, layered mounds of sediment trapped by biofilms. These formations appear in the record around 3.5 billion years ago, but they do not tell us exactly when oxygenic photosynthesis became the dominant mode of life. The genetic evidence is also limited. We can study modern cyanobacteria and work backward to estimate when their key innovations emerged, but the molecular clock is imprecise over billions of years. Then there is the long, puzzling plateau after the first surge. For over a billion years, oxygen levels hovered at just a few % of modern concentrations. Why did they not climb higher? What kept the planet locked in that low oxygen state for so long? The leading hypothesis involves a balance between oxygen production and consumption. The continents were still relatively small, and the oceans were rich in dissolved iron and other reactive elements that could soak up oxygen. Photosynthetic life may have been limited by the availability of essential nutrients like phosphorus and nitrogen, which are needed to build proteins and DNA. Without those nutrients, the cyanobacteria could not expand their populations enough to overwhelm the planetary buffers. It was a stalled engine, idling for a billion years until something finally broke the deadlock. That something is the second unanswered question. What triggered the Neoproterozoic oxygenation event around 800 million years ago? The breakup of the supercontinent Rodinia seems to have played a key role. As the landmasses drifted apart, new coastlines and shallow seas formed, creating vast new habitats for photosynthetic algae. The weathering of fresh rock on those new continents released nutrients into the oceans, fertilizing the growth of phytoplankton. At the same time, tectonic activity buried large amounts of organic carbon in deep-sea sediments, locking away the carbon that would have otherwise reacted with oxygen and removed it from the air. But these are all indirect clues. The exact chain of cause and effect is still debated. Some scientists argue that the evolution of the first green algae, which were more efficient at photosynthesis than cyanobacteria, was the crucial breakthrough. Others point to a drop in volcanic activity that reduced the supply of reducing gases. A few even suggest that a series of snowball Earth glaciations, which scraped nutrients off the continents and then released them in a pulse as the ice melted, provided the final push. Each hypothesis has its strengths, but none has been proven beyond doubt. The deeper mystery is why the entire process of oxygenation took so long. Photosynthesis evolved perhaps 3 billion years ago, but complex animals did not appear until around 600 million years ago. That is an enormous gap, a delay that still puzzles researchers. Was oxygen the only constraint, or were there other barriers, genetic, ecological, or environmental, that held life back? The fossil record hints that the first multicellular organisms may have evolved earlier than we think, but they remained microscopic and simple because they could not extract enough energy from the low oxygen environment to build larger bodies. Only when oxygen crossed a critical threshold did the door to complexity swing open. But what set that threshold, and why did it take so long to cross? These questions are not just academic, they touch on the fundamental nature of life's potential. If oxygen had risen faster, complex animals might have appeared 1 billion years earlier, and the history of life would be radically different. If it had risen slower or stalled permanently, Earth might still be a world of single cells and microbial mats. The timing of oxygenation may have been one of the great filters that determined whether intelligent life could ever emerge. And we still do not fully understand why it happened when it did. The search for answers continues in ancient rocks, in the genomes of living microbes, and in computer models that simulate the chemistry of an evolving planet. Each new study adds a piece to the puzzle, but the full picture remains tantalizingly incomplete. The story of oxygen is not yet a closed chapter. It is an ongoing investigation, and the most important questions may still be waiting to be asked. One of the deepest puzzles is why oxygenic photosynthesis evolved only once. If the ability to split water using sunlight was such a powerful advantage, why did no other lineage of microbes ever invent it independently? Every living plant and every cyanobacterium on Earth today traces that critical innovation back to a single common ancestor. That suggests that evolving the machinery to tear apart water molecules is extraordinarily difficult, far more complex than other forms of photosynthesis that use hydrogen sulfide or organic compounds instead. The reaction center proteins, the protective enzymes, the entire electron transport chain, all had to come together in just the right order, and any intermediate step would have offered little benefit while consuming energy. It may be that the path to oxygenic photosynthesis was so narrow, so chemically improbable, that even billions of years of evolution only stumbled across it once. That single accident, however, changed everything. Another mystery hangs over the long, quiet Middle Age of Earth, the period geologists call the Boring Billion. For roughly 1.5 billion years between the first Great Oxidation and the second surge, oxygen levels seemed to hover at just a few % of modern concentrations. What kept them there? The leading idea is that the planet's internal chemistry acted like a thermostat. Photosynthetic life pumped out oxygen, but volcanic gases, weathering rocks, and deep-sea sediments consumed it at roughly the same rate. The system was locked in a stable, low-oxygen equilibrium. But why did it stay that way for so long? Could it have been the scarcity of key nutrients like phosphorus and nitrogen, which limited the growth of photosynthesizers? Or was it the slow pace of tectonic change, which only gradually altered the balance between oxygen sources and sinks? Researchers are now using isotope geochemistry, analyzing the ratios of oxygen isotopes in ancient rocks, to trace how oxygen was cycled through the oceans and atmosphere billions of years ago. These measurements can reveal when and where oxygen was being consumed or buried, giving us clues about what held the system in check. Evolutionary simulations are also helping. By building computer models of ancient microbial ecosystems, scientists can test what happens when nutrient levels shift or when new metabolic pathways arise. The simulations suggest that the Boring Billion was not really boring at all. It was a period of slow, hidden experimentation, where oxygen remained low but life was quietly refining the tools that would later allow a second dramatic rise. The timing of that rise, the Great Oxidation Event itself, is another deep question. Why did it begin around 2.4 billion years ago and not earlier? The oxygen-producing cyanobacteria had been around for hundreds of millions of years before that tipping point, so what finally broke the chemical stalemate? One hypothesis points to a major shift in the composition of volcanic gases. As the Earth's interior cooled, the ratio of reducing to oxidizing gases released by volcanoes may have changed, gradually tipping the atmospheric balance. Another hypothesis involves the evolution of a new kind of cyanobacteria that was more efficient at producing oxygen, perhaps one that could survive in a wider range of environments. The rock record is frustratingly silent on this question. The most ancient fossils are rare and hard to interpret, but new techniques like analyzing the trace metal content of banded iron formations are giving us sharper tools. These metals act as fingerprints of ancient biological activity, and they may one day tell us exactly when and why the first great poisoning began. Every discovery in this field seems to open more questions than it answers. That is part of why the story remains so compelling. We are not simply looking back at a finished chapter. We are watching scientists piece together a narrative from fragments of rock and trace elements, using the latest technology to interrogate a world that vanished over 2 billion years ago. The next breakthrough could come from a drill core pulled from the Australian outback, or from a computer model running in a university basement, or from a new way of reading the chemical language locked inside ancient crystals. What seems certain is that the full picture of how oxygen transformed Earth is still being written, and the most important clues may be waiting in places we have not yet thought to look. That long chain of unanswered questions now reaches beyond Earth. If the rise of oxygen was such a rare and improbable event, even on a planet that was teeming with life, then what does it mean for our search for life on other worlds? The same gas that nearly sterilized our own planet is one of the most promising signals we look for in the atmospheres of distant exoplanets. Astronomers call it a biosignature, a chemical marker that could indicate the presence of living organisms. But the story of Earth's own oxygen history offers a sobering warning. Detecting oxygen does not mean we have found a friendly, complex world. It might mean we have found a world in the middle of its own Great Oxidation Event, a planet where a microbial catastrophe is unfolding, where the air is turning poisonous for the very life that produced it. When we scan the atmosphere of an exoplanet using telescopes like the James Webb Space Telescope, we are not looking for little green men. We are looking for imbalances in the chemical composition that cannot be explained by geology alone. Oxygen is a prime candidate because it is so reactive. On a lifeless planet, any free oxygen would quickly combine with rocks and volcanic gases, vanishing from the air within a few million years. To maintain a steady level of oxygen, something must be constantly producing it, and on Earth that something was life. But here is the catch. The oxygen we breathe today is at about 21%. That is a stable, high concentration that supports animals like us. But as we have seen, Earth's oxygen was not always at that level. It went through a long period of very low oxygen, then a sudden spike, then a billion-year plateau, then another spike. If an alien astronomer had observed our planet 2 billion years ago, they would have seen a world with a few percent oxygen, enough to indicate some sort of photosynthetic life, but not enough to suggest anything more than microbial mats. They would have been correct, but if they had observed Earth during the Great Oxidation Event itself, they would have seen a dramatic rise in oxygen, a burst that might have looked like a thriving biosphere. In reality, it was a planetary poisoning, a mass extinction in progress. So the presence of oxygen in an exoplanet atmosphere is not a straightforward sign of a complex, habitable world. It could mean we are looking at a planet in the middle of a biological revolution, or even a world where the oxygen level is too high, making it toxic for any large, active organisms. The context matters enormously. The atmospheric composition, the presence of other gases like methane or carbon dioxide, the planet's distance from its star, the age of the system. All of these factors must be weighed together to understand what oxygen really tells us. And there is another twist. Oxygen can also be produced without life, through the splitting of water molecules by ultraviolet light from a star. This process, called photolysis, can build up a small amount of oxygen in a planet's atmosphere, especially if the planet has lost its hydrogen to space. So a thin layer of oxygen around a rocky world might be a false positive, a sign of a dry, barren planet rather than a living one. Scientists are developing ways to distinguish between biological and non-biological oxygen. One key is to look for the presence of other gases that would be consumed by oxygen if life were not constantly replenishing them. Methane, for example, reacts quickly with oxygen. If both methane and oxygen are present in the same atmosphere, something must be producing them rapidly, and that something is often life. This is why the Earth's atmosphere was such a powerful clue for the early astronomers who first studied it from space. They saw oxygen and methane coexisting in a state of chemical disequilibrium, a signature that pointed directly to a living world. So when we search for oxygen on exoplanets, we are not just looking for a single gas. We are looking for an entire chemical story, a narrative that unfolds in the spectrum of starlight passing through an alien atmosphere. The story of oxygen on Earth, with all its twists and turns, is a guidebook for that search. It tells us that oxygen can be a sign of life, but also a sign of catastrophe, a sign of a world in transition, or even a sign of a dead planet lit by ultraviolet rays. Every detection will require careful interpretation, and the first few exoplanets with oxygen might not be the ones we expect. Perhaps the most exciting possibility is that we will find a world where oxygen is present at a level similar to Earth's during the Cambrian explosion, just high enough to fuel complex animal life, but not so high as to be toxic. That would be a world where the planetary Goldilocks zone has been achieved, and where creatures might be swimming in oceans that are just beginning to feel the pulse of a new kind of energy. But we will not know until we look. The search for oxygen in the universe is not just a technical challenge, it is a philosophical one. It forces us to reconsider what we mean by habitability, and whether the conditions that gave rise to us are common or vanishingly rare. The same molecule that once poisoned our own world may one day reveal that we are not alone, or it may remind us that the balance required for complex life is so delicate that it has only happened once in all the vastness of space. Either way, the story of oxygen is far from over. It is being written not only in Earth's ancient rocks, but in the light of distant stars. The oxygen we search for in exoplanet atmospheres is a double-edged clue. On one hand, it is one of the most powerful biosignatures we know, because free oxygen is so chemically hungry that it does not linger in an atmosphere unless something is constantly replenishing it. On Earth, that something was life, so if we detect oxygen around a distant world, it raises an exciting possibility that microbes or plants are actively pumping it out. But the story of our own planet warns us that this signal is not a simple green light. A world with oxygen might be teeming with complex animals, or it might be in the middle of a planetary crisis, a great oxidation event that is poisoning its own inhabitants. And what about worlds with no detectable oxygen at all? Could they still harbor life? Absolutely. For the first 2 billion years of Earth's history, life flourished in an oxygen-free environment. The oceans were filled with thriving microbial communities that never used or produced the gas. If an alien astronomer had scanned our planet during that era, they would have seen an atmosphere with almost no oxygen, and they might have concluded that Earth was barren. But they would have been wrong. So the absence of oxygen does not mean the absence of life. It simply means life of a different kind, the kind that does not rely on breathing the air we do. This realization forces us to broaden our definition of a habitable world. a planet might be teeming with microbes, even complex multicellular organisms, without a trace of oxygen in its sky. On the other hand, too much oxygen can be just as deadly as too little. We saw how high oxygen levels during the Carboniferous allowed giant insects to evolve, but also made the atmosphere more flammable and pushed the limits of oxidative stress. If a planet's oxygen concentration rose too high, it could overwhelm the antioxidant defenses of any large animal, effectively sterilizing the land and shallow seas. There may be an upper limit beyond which complex life cannot survive, even if oxygen itself is produced by photosynthesis. This narrow window, the Goldilocks zone of oxygen, becomes a critical filter when we assess the habitability of exoplanets. We cannot simply celebrate a detection of oxygen. We must ask how much and in what company. Is it accompanied by methane, indicating a biological source? Is it moderate, like Earth's 21%, or dangerously high? And what about worlds that have no oxygen at all? They may still be vibrant with life that we have not yet learned to recognize. The search for life beyond Earth is not just a hunt for familiar molecules. It is a lesson in humility, reminding us that the story of our own planet is only one chapter in a much larger book. Every new discovery will force us to rewrite that book, and the oxygen paradox will be at the center of the rewrite. The story of oxygen on Earth is not just a tale of ancient catastrophe and eventual triumph. It is a lesson in fragility. Every breath you take is a reminder that the conditions allowing complex life to exist are not guaranteed. They are the result of a delicate balancing act that took billions of years to perfect, and that balance can be disrupted. The same gas that powers your muscles could just as easily become a poison if its concentration shifts even slightly. That is the central paradox we have been exploring. A molecule that sustains us is also a molecule that can destroy us. And this paradox holds profound lessons for how we understand our own planet and our place in the universe. The first lesson is that complexity comes with a cost. Every advantage oxygen provided, more energy, larger bodies, faster metabolisms, came with a hidden price. Cells had to invest in antioxidant defenses. DNA had to be constantly repaired. Whole organ systems evolved just to manage the delivery and removal of this reactive gas. The very features that allowed animals to move, hunt, and think also created new vulnerabilities. There is no such thing as a free lunch in evolution, and oxygen was no exception. It gave life the fuel to build intricate bodies, but only if those bodies could also withstand the constant oxidative wear and tear. That wear and tear is why we age, why our cells slowly accumulate damage over a lifetime. It is the interest we pay on the energy loan that oxygen provides. The second lesson is about timing. The rise of oxygen did not follow a smooth, predetermined path. It was a series of stalls and surges, a billion-year pause, a sudden spike, another long plateau, and then a final burst that set the stage for animals. Each step depended on a confluence of geological and biological factors that could have turned out differently. If the Cyanobacteria had evolved a little later, or if the tectonic plates had shifted in a different pattern, the oxygen windows might have opened at a different time or not at all. The Cambrian explosion, the birth of all major animal groups, was not inevitable. It was a lucky alignment of planetary chemistry, evolutionary innovation, and sheer accident. That fragility should give us pause. It suggests that complex life may be rare in the universe not because it is hard to start, but because the conditions that allow it to flourish are so precisely balanced. A planet might have life for billions of years without ever crossing the oxygen threshold that allows animals to emerge. It might have too much oxygen or too little, or the rise might come at the wrong moment. The third lesson is about perspective. Oxygen was once a poison that nearly wiped out all life. Now we depend on it with every breath. That reversal took over 2 billion years. It is a reminder that our relationship with the environment is not static. What we consider essential or harmful today can shift over deep time. The microbes that first produced oxygen had no idea they were creating a new world. They were just surviving, and their waste changed everything. We are in a similar position today. Our own activities are altering the planet in ways we do not fully understand. The carbon dioxide we release, the nitrogen we fix, the plastics we manufacture, all of these are acting like the oxygen of the ancient Cyanobacteria, a byproduct that will reshape the Earth for millions of years to come. The lesson of oxygen is that life is a planetary force. It does not simply adapt to its environment. It changes that environment, sometimes with consequences that are catastrophic for the very life that caused the change. We are not separate from this cycle, we are part of it. The final lesson is one of humility. Our planet is the only world we know that has achieved this balance. The Goldilocks zone of oxygen, high enough for animals but low enough to avoid toxicity, has lasted for several 100 million years. That is a blink in geological time. It will not last forever. Eventually, the sun will brighten and the Earth will lose its oceans and the oxygen will fade away. But for now, we are living in a brief, fortunate window where the atmosphere is just right. Every time you take a deep breath, you are tasting that luck. The story of oxygen is not finished. It is still unfolding both on Earth and in the atmospheres of distant worlds. And the greatest mystery may be whether that delicate balance ever happens again. This entire story hinges on a single, strange truth. The world that was chemically stable, the one where oxygen was absent and life hummed along in a predictable, slow rhythm, was also a world that could never produce anything beyond simple microbes. It was a planet locked in a comfortable rut, a chemical equilibrium that had lasted for nearly 2 billion years. Every reaction, every volcanic puff, every microbial waste product, they all cancelled each other out. The system was balanced, but balanced at a low level. Energy was scarce, growth was slow, complexity was impossible. Then oxygen shattered that balance. It turned the atmosphere into a chemical roller coaster. The stability was gone, replaced by a constant churn of reactions, of oxidation, of new molecules forming and old ones breaking apart. From our perspective, that sounds like chaos, and in a way it was. But that chaos was also a release of enormous potential energy. Think of a log jam on a river. The water behind it is still, calm, stable, nothing moves. Then a single log shifts and suddenly the whole jam breaks free. The water surges downstream, churning and frothing. It is no longer stable, but that very instability is what allows the river to carve new channels, to carry sediment to new places, to power mills and sustain life along its banks. Oxygen did the same for Earth. It broke the log jam of chemical stability. In doing so, it unlocked a vast reservoir of energy that life could tap into. The poison became the fuel. The catastrophe became the opportunity. This is the deepest paradox of the entire story. The same molecule that nearly wiped out life also created the conditions for life to reach its highest potential. It forced organisms to adapt or die, and those that adapted gained a power that nothing before had ever possessed. The lesson is not that change is good or bad. It is that change is inevitable, and that life's greatest strength is its ability to turn a threat into a resource. The anaerobic microbes that fled to the deep sea refuges did not fail. They chose survival over transformation, but the ones that stayed near the surface, the ones that weathered the poison and learned to breathe it, those were the ones that opened the door to everything that followed. They turned a world that was chemically hostile but predictable into one that was chemically unstable but bursting with possibility. We are the descendants of that gamble. Every animal, every plant, every fungus carries the signature of that ancient choice, to embrace the poison and make it a partner. So when you look back at the Great Oxidation Event, do not see it only as a disaster. See it as a reset, a clearing of the old world that allowed a new one to take shape. The stability of the anaerobic age was a prison as much as a sanctuary. The instability of the oxygen age was dangerous, but it was also liberating, and liberation always comes with a price. Every modern convenience carries the invisible cost of oxygen. The fuel that burns in your car engine, the fire that heats your home, the electricity that lights your street, each one is a chemical marriage between carbon and the very gas that once poisoned our world. We have built an entire civilization around the same reaction that powers every cell in your body, the combination of oxygen with fuel to release energy. But where our cells do it slowly and carefully inside mitochondria, our engines do it fast and hot, consuming oxygen at a rate that would have been unimaginable to the ancient microbes that first learned to tolerate the gas. Our power plants, our factories, our vehicles, they are all machines for oxidation. They take carbon from fossil fuels and use oxygen from the air to produce heat, motion, and light. In doing so, they release carbon dioxide, a gas that was once abundant in the early Earth but was gradually locked away by ancient life. Now we are returning that carbon to the atmosphere at a speed that far outpaces the natural processes that once regulated the planet's chemistry. This is a new kind of experiment, one where oxygen is not the waste product but the consuming agent. And just as the Cyanobacteria changed the world without understanding the consequences, we are altering the atmosphere in ways that will echo for millennia. Oxygen is not just the breath of life. It is the partner in every flame, every explosion, every engine that drives our modern existence. The same molecule that enabled the rise of animals now enables the rise of a technological world. But that world depends on a delicate balance. We rely on a steady supply of oxygen in the air, currently at 21%, a level that has remained remarkably stable for hundreds of millions of years. Yet our activities are shifting the composition of the atmosphere in other ways, adding carbon dioxide and other gases that could, over long timescales, alter the very chemistry that sustains us. We are living in an oxygen-dependent civilization, and that dependence is both a gift and a responsibility. It is a gift because oxygen provides the energy density that makes our technology possible. It is a responsibility because we must ensure that we do not disrupt the planetary systems that maintain that oxygen at a livable level. The story of oxygen is not over. It continues in every breath we take, in every fire we burn, in every decision we make about how to power our future. And the ancient lesson still applies. The same molecule that enables life can also threaten it if the balance is lost. We are not separate from that story. We are its latest chapter. Every breath you take is a chemical treaty signed billions of years ago. That gentle rise and fall of your chest, the almost invisible exchange of gases in your lungs, it is a direct link to the ancient catastrophe that nearly wiped out all life. The oxygen you inhale is the same kind of molecule that once tore apart the cells of helpless microbes. But now, instead of a poison, it is the fuel that keeps your heart beating, your brain thinking, your muscles moving. That transformation did not happen overnight. It took 2 billion years of evolution, extinction, and adaptation to turn a global poison into the very substance we depend on with every moment of our lives. But the legacy of that ancient war is still written inside your body. Every cell in your body contains mitochondria, the tiny power plants that burn oxygen to produce energy. And just like the first microbes that learned to handle the gas, your cells must constantly defend themselves against the same reactive oxygen species that once caused so much damage. This is where the story becomes deeply personal. The same process that gives you energy also produces tiny, volatile fragments that can damage your DNA, your proteins, and your cell membrane. Your body has inherited the same antioxidant enzymes that evolved billions of years ago, superoxide dismutase and catalase, still working tirelessly to neutralize those sparks before they start a fire. When those defenses begin to weaken, or when the production of reactive species outpaces the body's ability to handle them, a state called oxidative stress sets in. This is not a vague concept. It is a measurable imbalance that has been linked to many of the health challenges we face as we grow older. The gradual accumulation of oxidative damage in your cells is one of the underlying factors in aging itself. It is also connected to a range of conditions that affect the heart, the brain, and other vital systems. Scientists now understand that the very mechanism that allowed complex life to emerge also imposes a kind of built-in cost, a biological tax that we pay as time passes. The same gas that gave us the energy to build civilization also slowly wears us down from the inside. Understanding this history helps us see our own bodies in a new light. We are not just random collections of cells. We are walking archives of a planetary drama that unfolded over billions of years. The air you are breathing right now contains oxygen that was produced by ancient microbes, consumed by early animals, and recycled through countless generations of life. The atmosphere that surrounds you is not a static blanket. It is a dynamic, fragile balance that has been maintained by the living world for hundreds of millions of years. And that balance is more delicate than we often realize. A shift of just a few % in oxygen concentration, either higher or lower, would be catastrophic for most complex life. We are living in a narrow window of habitability, a Goldilocks zone that our planet has managed to sustain through a combination of geological and biological feedbacks. Oxygen, then, remains a double-edged sword. It is the reason you can run, think, and dream, but it is also the reason your cells slowly accumulate damage over a lifetime. It is the gas that fuels your existence and the same gas that will one day contribute to your decline. This is not a dark thought. It is a humbling realization that every advantage comes with a cost, and that the conditions that allow complex life to flourish are both precious and precarious. The next time you take a deep breath, let it be a reminder of that ancient transformation and of the delicate balance that keeps you alive right now in this very moment. Without that ancient cataclysm, life would never have grown beyond a microscopic film. The entire planet would have remained a vast, quiet expanse of single cells, drifting in anoxic oceans and clinging to volcanic vents. There would be no green leaves rustling in the wind, no birds cutting through the sky, no animals grazing on grassy plains. The world would be silent in a way that is almost impossible for us to imagine. The only sounds would be the hiss of steam and the rumble of tectonic plates. The only colors would be the rusty red of iron-stained waters and the pale haze of a methane-rich sky. The atmosphere itself would be thin and poisonous to us, laden with hydrogen sulfide and carbon dioxide. There would be no ozone layer because ozone requires oxygen to form. Without that protective shield, ultraviolet radiation from the sun would bathe the surface, destroying any exposed organic molecules and making the land completely barren. The continents would be rocks, bare and lifeless. The oceans would be warm and murky, filled with microbial mats that stretched for kilometers, but nothing more. No complex cells would have evolved because the energy budget would have been too small. Without the mitochondrial partnership, cells would have remained simple, bounded by a membrane and a few internal structures, but incapable of building the machinery needed for multicellular bodies. There would be no eyes to see, no ears to hear, no nervous systems to process information. The world would be a place of slow, steady chemical exchanges, a beautiful but static system that had reached its peak 3 billion years ago and never changed again. Perhaps some forms of multicellular life might have emerged using different metabolic strategies, but they would have been thin, flat, and passive, never developing muscles or bones. They would have drifted like living sheets, absorbing nutrients from the water, never moving with purpose, and they would never have produced intelligence. No creature would have looked up at the stars and wondered about its place in the universe. The Earth would have been a living world, but a silent one, a world without ambition or curiosity. The very concept of technology, of fire, of written language, of art, would have never arisen because there would have been no minds to conceive them. The oxygen revolution was not just a biological event. It was the unlocking of a door that led to consciousness itself. Without that first poisoning, without the long struggle to turn a toxin into fuel, the planet would have remained a beautiful but empty cradle, a place where life existed but never truly awakened. So the next time you look at a blue sky or hear a bird sing or feel the breeze on your skin, remember that you are witnessing the aftermath of a catastrophe. The world you know is a world that was forged in crisis, and every complex thing you see is a testament to life's ability to transform poison into possibility. That lush world almost never came to be. Somewhere in the branching paths of deep time, the oxygen revolution could have stalled. The cyanobacteria might have never evolved, or the planetary buffers might have held forever. Imagine for a moment an Earth where free oxygen never accumulated beyond a trace. The air would be thick with methane and hydrogen sulfide, a faint rotten egg smell carried on a stagnant breeze. The sky would not be blue but a hazy orange or pale brown, colored by organic haze and volcanic dust. No ozone layer would form, so ultraviolet radiation would bathe the surface, making the land a sterile desert where no complex molecule could survive for long. The oceans would be warm and chemically rich, stained green or rust-colored by dissolved iron and sulfur. Life would exist in vast microbial mats, layered communities of anaerobes spreading across the seafloor like living carpets. They would feed on hydrogen, sulfur, and carbon dioxide, releasing waste products like methane and acetate. But no cell would ever grow larger than a few micrometers. No nucleus would ever evolve because the energy budget would be too tight. No mitochondria could ever form because there would be no oxygen to harness. The world would be quiet, almost silent, except for the hiss of volcanic vents and the gentle lap of anoxic waves. No leaf would rustle, no bird would sing, no foot would ever press into soft soil. The land would remain bare rock and dust, untouched by roots or burrows. For billions of years, this pattern would repeat, a steady, slow cycle of microbial growth and decay, with no arrow pointing toward complexity. The planet would be alive, teeming with trillions of organisms, yet utterly empty of anything we would recognize as a dynamic, thinking world. No creature would ever look up at the stars because there would be no eyes to see them. No mind would ever wonder about the origins of life because there would be no mind at all. That version of Earth is not a fantasy, it is the most probable outcome. Given the chemical odds, given the fragility of the oxygen pathway, the balance could have tipped the other way. The fact that it did not, that oxygen rose and stayed within a habitable range, is a reminder of how contingent our existence really is. Every breath you take is a lucky break in a chain of accidents stretching back 2 billion years. The next time you stand under a blue sky, let that color remind you of the thin line between a world of silent microbes and a world of forests, cities, and songs. That thin line is the only thing separating us from a silent planet, a world where life never broke free from its microscopic prison, where the oceans held nothing but microbial mats and the land remained bare rock under a hazy sky. But that is not the world we live in. We live in a world where a poison became a breath, where a gas that nearly erased all life instead became the fuel for forests, cities, and songs. The story of oxygen is not merely a chapter in Earth's history. It is the story of how life learned to transform a threat into an opportunity, and how that transformation opened the door to everything we know. Every cell in your body carries the signature of that ancient gamble. Inside your mitochondria, the same chemical reaction that once tore apart helpless anaerobes now powers your thoughts, your movements, your very existence. The reactive oxygen species that your cells work tirelessly to neutralize are the direct descendants of the same volatile fragments that caused the Great Oxidation Event. You are a walking archive of a planetary crisis that happened 2 billion years ago, and the air you breathe is the same kind of air that allowed the first trilobites to crawl across the seafloor, the first fish to swim, the first reptiles to crawl onto land, and the first humans to look up at the stars. That progression was never guaranteed. It depended on a series of accidents, a broken buffer, a stolen bacterium, a long frozen pause, and a final surge of oxygen that pushed the planet into a new state. Every step could have failed, every threshold could have remained uncrossed, but they did not, and here we are, living in a brief, fragile window where the atmosphere holds just enough oxygen to sustain complex bodies without overwhelming them. That window will not last forever. Eventually, the sun will brighten, the oceans will boil, and the oxygen will fade away. But for now, we are here, and we are the product of that ancient war. The poison that once wiped out nearly all life has become the very substance that allows you to take a deep breath, to feel the wind on your skin, to wonder about the universe beyond your planet. That transformation is the most profound lesson of Earth's history. It teaches us that even the most destructive forces can, under the right conditions, become the foundation for something beautiful. It reminds us that life does not simply adapt to its environment. It reshapes that environment, sometimes with consequences that are catastrophic, and sometimes with consequences that are miraculous. And it asks us to consider that we, too, are part of that ongoing story. The oxygen you are breathing right now was produced by ancient microbes, consumed by countless generations of animals, and recycled through the living world for billions of years. You are not separate from that history. You are its latest expression. Every breath is a link to a time when the sky turned blue, when the continents turned green, when the first eyes opened, and when the first mind began to ponder its own existence. That is the gift of oxygen, a poison that became a partner, a catastrophe that became a cradle, and a story that is still being written with every breath you take. The planet you stand on today was forged in that very contradiction. A waste product from simple bacteria became the most transformative force in Earth's history, first as a poison that nearly erased all life, then as the fuel that made every complex creature possible. The survivors of that ancient catastrophe did not merely endure the poison. They learned to harness it. They built antioxidants to neutralize its fury, then flipped the switch and began using it to extract staggering amounts of energy from the same food their ancestors had barely survived on. That stolen energy allowed cells to grow larger, to carry more genes, to form partnerships that created the first complex cells, and eventually to build bodies that could move, sense, and think. Every animal that has ever lived, every bird that has flown, every fish that has swum, every human that has ever looked up at the stars owes its existence to that single, improbable transformation. Oxygen went from being a planetary assassin to the very currency of complex life, and that journey holds a mirror up to our own time. We live in an age of unprecedented power, built on the same principle of turning a resource into energy. But just as oxygen once threatened the world that produced it, our own innovations carry hidden costs. The fires we burn, the engines we run, the very civilization we have built, all depend on the same kind of chemical reaction that first poisoned the Earth. The lesson is clear: every gift comes with a responsibility. Every breakthrough carries a shadow, and the survivors of the next great transformation will be those who learn to balance that power with wisdom. If this story has given you a new perspective on the air you breathe, consider sharing it with someone who might appreciate it too. Hit the like button if you want more deep time narratives. Subscribe so you do not miss the next chapter, and leave a comment letting us know what other ancient mysteries you would like us to explore. Because the past is never really past. It is still breathing inside you with every rise and fall of your chest. And the question it leaves us with is this: what will we do with the power that oxygen gave us, and will we learn from its double-edged nature before it is too late?